Hello பசங்களா, welcome all of you to We Need Tamil Nadu. எப்படி இருக்கீங்க? நல்லா இருக்கீங்களா? ஸோ இன்னைக்கு வந்து நம்ம செல் த யூனிட் ஆஃப் லைஃப், ஓகே, அந்த சாப்டர் வந்து டீகோட் பண்ண போகிறோம் வித் என்சிஆர்டி லைன்ஸ் as well as notes as well as GM express, GM diagrams. ஸோ ஒரு காம்பினேஷன் தான், ஓகே, ஒரு கிச்சடி மாதிரி we will be doing everything, okay, NCRT line by line, notes, PPT is there, plus your diagrams are going to be there, plus my one shot notes also is going to be there. So are you all ready? With that we will also have 3D as well, okay, so you will understand everything, you look at everything, and also you will understand that okay, this chapter was this easy, because cell the unit of life and the students வந்து ஐயோ மேம் முடியாது மேம், அந்த சாப்டர் வந்து ரொம்ப கஷ்டம் மேம், முடியாது மேம், அந்த மாதிரி ஒரு ஃபியர்ல இருக்கு. ஸோ அந்த சாப்டர் வந்து நம்ம டீகோட் பண்ணலாம். ஸோ இஃப் யூ ஆர் ரெடி ஃபார் த கிளாஸ், quickly put a lot of fire emojis in the chat box and make sure that you're subscribed and smashing the like button. Make sure that you like the video from everyone's phone, neighbor's phone, next person's home, friend's home, every home you have to go and make sure that you smash the like button as well. So start பண்ணலாமா students? So இதுதான் நம்ம செல், okay ma'am, எவ்வளோ சிம்பிளா இருக்கு மேம் பார்க்கிறதுக்கு, பட் ஆனா so many organelles we have to study, right? Yes, so this is your cell, okay, this is the beautiful nucleus that we keep drawing. This will get a little stuck students because இது வந்து free site தானே, so அதனால வந்து it will get little stuck, but doesn't matter, it will work. So this is every small thing we have to study about the entire organelle, okay, everything we have to study. So look here, now it will take some time to load, but this is the beautiful nucleus that we always study. Nucleolus will be there inside that, nucleus will be there. So adukapram, see, nucleus is there, yes ma'am, see, nucleus I saw. This is what we always talk about, right? We talk about Golgi apparatus, we talk about your mitochondria. This is your mitochondria, which is the powerhouse of the cell. So adu madri lot of organelles we are going to study, but ana or introduction venume chapter ku, yes ma'am, introduction mukkiyam ma'am, okay, introduction is very mukkiyam. So today we are going to do the introduction and we will understand the entire flow of the chapter. Students, நம்ம வந்து cell பத்தி பேசும்போது I feel like every student, like let's say even if I ask a commerce student about cell, he is going to tell me it is the structural and functional unit of life. And second thing that he knows in science is mitochondria is the powerhouse of the cell. So இது மாதிரி ரெண்டு வார்த்தை every students know, right? Commerce students ஆகிட்டும், art students ஆகிட்டும், any students, they know these two lines, that cell is the structural and functional unit. So don't you think we also should know this? Okay, it is the structural and functional unit of life. நம்ம எப்பவுமே students unit word use பண்ணும்போது unitனா நம்ம automatically we should understand that repeating, okay, unitனா என்ன, repeating, okay. இந்த board வந்து ரொம்ப வலுதா இருக்கு, so அதனால I feel like I'm a small, one small like one chinchan in front of it, okay. So cell is the structural and functional unit. When I say cell, what do you understand, ma'am? It is very small, ma'am, and anything and everything has a cell, right? So students, normally also nama animal tissues la vanda we have studied that there will be cells, okay, there will be cells. These cells will accumulate to form tissues, correct dane ma'am, okay, the cells will accumulate to form tissues, illana we will have. So idu mari normally if I tell you draw a cell, you will draw a cell like this, okay. This is the simplest cell any person can draw. With this cell, okay, with this cell an accumulation forms a tissue, okay, accumulation forms a tissue, okay. This is the cell, correct dane. Now this tissue will accumulate to form what students? Nama organ, okay, organ kaparam organ system, அதுக்கப்புறம் ஒரு complete organism, அதுக்கப்புறம் ஒரு complete organism, correct தானே? Yes ma'am, perfectly right, okay, perfectly right. So இதுதான் நம்ம flow chart, இதுதான் நம்ம basics of the cell chapter. Now students, when I tell again cell ma'am, students, you are going to listen to cell a million times, okay. Cell na நம்ம normally on the basis of how many cells are there, நம்ம வந்து ரெண்டு கேட்டகரியில டிவைட் பண்ணணும். Unicellular organism, okay, unicellular organism, அதுக்கப்புறம் multicellular organism, okay, multicellular organism, okay. நம்ம unicellular organism na uni na என்ன students? uni na என்ன? இந்த uni word அர்த்தம் என்ன? uni na ma'am single. Okay, uni na enna single, right? Uni means single. So in the unicellular organisms means these people have a single cell. Correct? These people have a single cell or single cell organism. Example which is coming in your mind, all of you tell me quickly in the chat box. Ma'am, when I tell about unicellular, the first thing that comes to my mind is amoeba. Correct? Then, so we know that there is amoeba, which is an irregularly shaped organism which has pseudopodia, and the pseudopodia will be the one that is going to help in the movement. Contractile vacuole is there. So in the single cell organism, most important ஆன ஒரு விஷயம் என்னன்னா, this fellow will not have division of labor. Division of labor na example என்ன, இப்போ chat boxல இவ்வளோ students இருக்கு, எல்லா students கிட்டும் I'm telling students, make sure that you know one person is looking at the attendance, one person is making sure that all the students are subscribing, one student is making that there are no spammers. So அது மாதிரி division of labor, I have given each job to one dedicated person. அதுதான் division of labor. But ஆனா நான் சிங்கிளா இருக்கு மேம், சிங்கிள் செல் தான், சிங்கிள் செல்ல division of labor கிடையாது. excretion ஆகட்டும், reproduction, circulation எல்லாமே இந்த single cell da perform pannum. So single cell means you should remember that there is no division of labor. No division of labor. Okay students, no division of labor. Ana இந்த சைடுல நமக்கு என்ன இருக்கு, multinational companies, என்ன multi cuisine hotels na என்ன ma'am, Asian food will be there, continental food will be there, அது மாதிரி different types of food will be there. So multi na என்ன ma'am, multi na students there will be many cells. Correct தானே? There will be many cells. We know so many examples, right? We know so many examples. Okay, we are there, animals are there, mammals are, everything we have. So many cells, and here students there is division of labor. Is present. இவ்வளோதான் introduction of the chapter. இவ்வளோதான் introduction of chapter. Okay வா? Single cell, no division of labor, many cells, division of labor. So இங்க வந்த students mainly என்னன்னா, எல்லா செல்ஸுமே it will take up a responsibility. Now in the chat box some of you are telling me, yes ma'am, I will make sure that everyone smash the like button, I will make sure that everyone subscribe to the channel, I will make sure that no one talks unnecessary in the chat box. அந்த மாதிரி ஒரு division of labor நடந்தேன்னா my work is very easy, I just have to teach. But imagine I was a single cell, எல்லாமே I should only do, I have to manage that, I have to manage this, I have to manage this. In the channel start பண்ணும்போது I was a single cell. Correct. I was only doing the videos, I was only doing the live, I was only doing the shorts, I was only managing the channel. அது மாதிரி ஒரு single cell la இப்போவுமே I only manage the channel. But ஆனா the thing is now we have one more teacher to do physics. அது மாதிரி in December starting chemistry teacher will come. So we will, division of labor will happen. Now they will take care of their subject, I will take care of all of your responsibility, what you need, what mentoring you need. அது மாதிரி ஒரு responsibility. But ஆனா it is difficult to be a single cell. But one more thing you should understand. See, if I am telling you that I am doing videos, I am doing live, I am doing everything, you should understand that okay, ma'am is capable of doing it, right? So, அது மாதிரி தான் இந்த cell வந்து students, how can a single cell வந்து எல்லாமே perform பண்ண முடியும்? That means this fellow, this single cell, okay, is a independent entity. It is what? It is independent. Okay, it is independent. So, independent na, that means it doesn't care about everything else. It is like, I will do my job properly. If you want, you take care of your job. அது மாதிரி ஒரு attitude இருக்கும். I am independent. Okay, light வந்து ரொம்ப reflect ஆகுது students. 1 second. இப்போ ஓகேயா? Yes, okay. I don't know which light is reflecting where, but ஆனா try பண்ணி check பண்ணலாம். Okay, yes. So, it is independent. It is independent means what? Yella organelles also, students, any organelles you take for that matter, yella organelles me oru dependency irukum. Okay, everything in our body has a dependency to something else. But ana in the cell mattum da, okay, independent existence. That means it doesn't care who is doing what is happening. I can do everything myself. So, adanala da single cell organism is able to function properly. Makes sense, no? Makes sense. No? If that person is not capable, I'll never give them a task. Adu madri tha. Same. Okay. So this is understood. This is the basic of our beautiful chapter, Cell, the Unit of Life. கொஞ்சம் அதிகமாக தப்பாச்சு. Cell the unit of life. Clear? Okay? Done. Okay. Now we will start learning the introduction part. Okay? இது மாதிரி notes இருக்கும் students, so you don't have to worry. It is the smallest functional unit which all the functions of the body is carried out. Anything less than, okay, listen here carefully, anything less than a complete structure of a cell does not ensure independent living. That means even if you are saying that mitochondria is the powerhouse of a cell or any organelles for that matter students, அது வந்து கம்ப்ளீட் செல் கிடையாது. ஸோ அதனால அதுக்கு டிபெண்டன்சி இருக்கும். பட் ஆனால் ஒரு செல்லுக்கு வந்து டிபெண்டன்சி இருக்காது. ஸோ இட் என்ஷூர்ஸ் இண்டிபெண்டன்ட் திங். ஸோ like wall is made of bricks, similarly all living organism is made of cells. So you look here, one cat is there, if I take a magnified image I will see animal cells. Plant is there, I will see plant cells. So adu madri tha, cell is present everywhere. Okay, now life starts with a simple zygote and finally turns into a complex multicellular organism. Simple ana human reproduction la enna we have understood students, namakku vandu sperm irukku ma'am. In the sperm vandu male gamete, adu vandu ovum ova கிட்ட fuse ஆகணும். இந்த ova and sperm will fuse together and it will form your zygote. Correct தானே students? இந்த zygote வந்து implantation அப்புறம் that gestation period அப்புறம் we are going to get a fetus. Okay, you're going to get a fetus. This is the basics. So they are saying that life starts with a simple zygote and finally turning into a complex multicellular organism. Correct thane? Normally nama blastular stage, modular stage at the stage kapram blastocyst implantation nadakum. So on the entire process la simple zygote in start agudhu or simple process da ida. This is because single cell keeps on dividing and forms more complex multicellular organism. So where does this entire origin happen? It happens from a simple zygote dividing multiple times and forming a multicellular organism. Okay, so you can see from zygote we are going to a 2-cell stage, then the fetus formation completely happens. Now on the basis of complexity, எவ்வளோ காம்ப்ளெக்ஸாக இருக்கும் ஆர்கானிசம், பேசிஸ் ஆன் தட் வி ஹவ் யூனிசெல்லர் அண்ட் மல்டிசெல்லர் ஆர்கானிசம். யூனிசெல்லர் ஆர்கானிசம் எக்ஸாம்பிள் வந்து அமீபா இருக்குது, பாக்டீரியா இருக்குது. மல்டிசெல்லர் ஆர்கானிசம் வந்து ஹைட்ரா, டாக், மேன். ஓகே, இங்கே வந்து யூனிசெல்லர் ஆர்கானிசமில் என்ன இருக்கும் ஸ்டூடெண்ட்ஸ்? இண்டிபெண்ட் எக்ஸிஸ்டன்ஸ். ஓகே, என்ன இருக்கும்? இண்டிபெண்டான எக்ஸிஸ்டன்ஸ் இருக்கும். அந்த செல் வந்து இண்டிபெண்டாக எல்லா வேலையும் இட் கேன் டூ. It can do it by itself. Okay, and here you have no division of labor. Single cell performs the essential functions of life. Essential functions of life means what? Be it circulation, okay, be it circulation, be it excretion, நம்ம life processல எல்லாமே. Okay, be it reproduction, okay, be it respiration, everything this cell will perform by itself. Okay, clear to all of you? Is it visible? Yes ma'am, it's visible. Yes, okay, got it. Circulation, excretion, reproduction, respiration, ellame, bacteria, amoeba, paramecium, all the cells are there. So here independent existence you have understood. Now look here, multicellular organisms have many cells and they do exhibit division of labor. They have division of labor. Cells are specialized to perform specific function. இப்போ வந்து நார்மலா பார்த்தேன்னா students, நம்ம circulatory system வந்து it will perform what? Ma'am, it will perform circulation. Okay, circulatory system வந்து it will perform what? It will perform circulation. Correct தானே? Yes ma'am. Okay, அது மாதிரி நம்ம I'm so sorry. Yes students, okay, so circulatory system வந்து circulation perform பண்ணும், respiratory system வந்து respiration perform பண்ணும். Okay, respiratory system வந்து respiration perform பண்ணும் ma'am. So எல்லா சிஸ்டமுமே division of labor is there students. See each other. Now can respiration do circulation? No. But that movement of gases, everything they will take care. So அது மாதிரி தான் எல்லா சிஸ்டமுமே they are individual. They know what they have to perform. They will not interfere with each other's job. Like that I will do this, I will do this. அது மாதிரி ஒரு mentality கிடையாது. They will do the proper. Why? Because division of labor is there in multicellular organisms. If students cells are performing this, you understood. Okay. We are multicellular organism. We have organs with specialized cells performing specific functions, right? We have liver, lungs, thyroid, brain, heart, stomach, kidney, bladder, everything we have. Each of it has its own job as well. Okay. Now, look here. Okay. First, we are going to understand cell discovery. How, ma'am? Who exactly found the cell? Okay. So, students, cell were first discovered by Robert Hooke. Okay, Robert Hooke, who is an English scientist in 1665. Okay, in the dates ellame it's important. Robert Hooke observed thin slice of cork. Okay, cork under his designed simple microscope. So ena and the tree bark illa and the cork in remove panni he was observing. But ana or important ana vishayam enna Robert Hooke vanda he noticed the dead cell. Okay, he analyzed what, students? He analyzed the dead cell, not the living cell. Okay, but the discovery happened because of Robert Hooke, where he took a thin cork slice and he saw it under a simple microscope. Okay va? See, this was how the microscope was before. இப்போ வந்து நம்ம simple system தானே, but ஆனா அந்த டைம்ல அது மாதிரி simple system கிடையாது. So அந்த systemல he took a small cork because everything has cell. So adanale anything you take you would have seen a cell, but ana he observed a dead cell. He did not observe a living cell. Okay va? Next one, students, see, he noticed partitioned boxes. So when he was noticing this, no students, he noticed that they were like little rooms. Okay, idu madri honeycomb shape little rooms madri irunduchu in the cells vanda oru compartments madri. Okay, idu madri compartments madri he observed it. Okay, so he's saying that he noticed partitioned boxes or compartments separated by walls. So that's what compartments separated by walls. Okay, idudha nama cork. Okay, idudha nama cork. So he took it under the microscope. He observed it like this. The compartments reminded him of a small room and he gave the name called as cellulae or cells. Okay, cell or cellulae in Latin means little room. So because of this, okay, because of this he felt like they were like compartments. Okay, compartments with small like partitioning, students. Okay, compartments with proper partitioning. So he called this compartments as what? Cellula. Okay, lay also you can tell, cellulae, which are actually cells. Now we call it as cells, right? We do not call it cellulae, we call it cells. Okay va? Now look here, this is how it was looking. The cells that Hooke observed were actually dead cells. Okay, it was dead cells from the bark of the cork, okay, tree. He explained his observation in a book called as Micrographia. So in that book he mentioned, so from a tree he took a bark, from the bark he noticed it, and what he observed was dead cells, and they were what? They were looking like honeycomb. Okay, they were looking like honeycomb. In the notes ellame description box la irukum. Also join our telegram, we need Tamil Nadu telegram, and the telegram வந்து last minute la lot of help it will happen, where I will share you a lot of DPPs and other things. Okay, so make sure that you're part of it. Also the cells that he observed resembled like a honeycomb. Yes or no? Don't it look like a honeycomb, students? Yes ma'am, it does look like honeycomb. So barkina he extracted and he saw dead cells. So question can come, Robert Hooke observed dead cells or living cells? So answer should be dead cells because he took it from the cork. So first discovery is clear to all of you? Yes or no? First discovery is clear. Can we go to the next discovery? So these notes are very good. Okay, these notes are made by our Vedantu team itself, so you don't have to worry. Okay, we use this notes sometime in our batch classes as well, so you can take full help of this notes. Okay, understood? Huh. In 1674, Antony van Leeuwenhoek, a Dutch scientist with an improved microscope, studied living cell, that is bacteria, protozoa, RBC, etc. for the first time. Students, in the Antony van Leeuwenhoek வந்து நம்ம ஒரு ஃப்ரெண்ட் இருக்கும்ல கிளாஸில் who is curious about everything, let's say he sees a little mud, he will start rubbing and seeing what is there in that. Let's say he sees something shining, he'll take that and அது மாதிரி ஒரு friend, if you have that friend, please let me know in the comment section. So அது மாதிரி ஒரு friend தான் Antony van Leeuwenhoek. He was curious about everything. எது பார்த்தாலும் he used to take his microscope and go and observe. Okay, it was more advanced, it was not like proper electron microscope, but ஆனா advanced இருந்துச்சு. So what he used to do, ஒரு pondல water இருந்துச்சுன்னா he used to use the microscope. Blood stain he sees somewhere, he used to use the microscope. So அது மாதிரி he found out that there is actually living cells. So here you can see he observed pond water, blood, okay, blood, thin slices of plant material, okay, thin slices of plant material, animal muscles, etc., and could see tiny, very fast moving bodies. He called them animalcules, okay, which means that little animals in Latin, because அது வந்து மூவ் ஆகுமே, நம்ம மைக்ரோஸ்கோப்பில் பார்க்கும்போது இந்த செல்ஸ் வந்து மூவ் ஆகும். ஸோ அது மூவ் ஆகிறதுக்கு he told, okay, maybe these people are tiny animal cells, okay, they are moving everywhere, so little animals. But Antony van Leeuwenhoek was the person who first noticed the living cells, okay, living cells. Clear? This way, if you learn also, students, you are going to understand everything. Later I will also draw and teach you. I will also make you like one shot notes as well, so don't worry, make sure that you are understanding everything and smashing the like button. Okay, yes. So he was the one who noticed the living cells, and he noticed it where, students, in the pond water. Okay, he noticed RBC in blood. Okay, he noticed thin slice of plant cell. எல்லாமே he was going with the microscope. Okay, Antony van Leeuwenhoek. Okay, now look here. Next was the discovery of cell. Robert Brown discovered the presence of nuclei in the orchid root. So இப்போ வந்து நமக்கு two discoveries நடந்துச்சு, students. Robert Brown and Antony van Leeuwenhoek. இப்போ வந்து நமக்கு Robert Hooke and Antony van Leeuwenhoek. இங்க வந்து Robert Brown. So three discoveries. Robert Hooke and Antony van Leeuwenhoek, both of them found cells, but Anna Robert Hooke discovered the cell which was actually dead cell, which was extracted from the cork. Okay, second one was the Antony van Leeuwenhoek. He worked on living cells. He noticed it in a pond of water, in blood and all of this. He noticed the moving cells and he called them little animals. Okay, adukapram came Robert Brown saying that in the cells சென்டர்ல வந்து ஏதோ இருக்கு, அதுதான் நம்ம நியூக்ளியஸா இருக்கும். So he noticed the presence of nucleus in orchid's root. Okay, இதுதான் orchid, அது rootல he noticed that every cell has some dot like structure right at the center, and that would be called the nucleus. Okay, is this clear? Is this clear? It is visible, right? If you want, I can reduce the brightness little more. Okay, little more I can reduce. 1 minute, students. Okay, might be little, I'll be little dark, but Anna sacrifices for you people. Okay, see, I'll look dark. Now it's more visible, right? Okay, done? Understood this? Yes, ma'am, understood. Easy. So this we will come back later. Okay, this we will come back later. So right now I'm writing a flowchart for all of you. Discovery. So discovery you have to 1, 2, 3. Okay, first one, who, students? Robert Hooke. Okay, Robert Hooke. Robert Hooke, what did he find? Ma'am, Robert Hooke was the person who found out cell. Okay, he discovered cell. Okay, and named it cellulae. Okay, cellulae na little rooms, okay, which are called little rooms, and they were honeycomb shaped. Okay, discovered cell and he named it cellulae or else cell, which are little rooms which were looking like the honeycomb type structures. Okay, okay, this is how it was present, students. And he worked on cork, okay, of a tree bark. Okay, so he technically found what? Dead cells. What he noticed were dead cells. This is how you should write your notes also. Everything that you need should be there in your notes, but at the same time, it should be short notes. Okay, wow. It should be short notes. Students, I don't like this line, so we will make it beautiful. Beautiful. இது வந்து height கிடையாது students, so அதனால I should climb up and write. Okay, but I'm not that short also. But ஆனாலும் board is too above. Yes. Oh. Yes. Okay, wow. Let's try and fix the line. This line is looking at this line and laughing. Okay, what are you, bro? Why are you not even standing straight? Okay. Okay, somewhat straight lines. Okay, better than what I drew before. So you know, no, I can't draw straight lines. But ஆனாலும் நம்ம attempt பண்ணணும். Okay, என்ன பண்ணாலும் plan பண்ணி தான் பண்ணணும். Okay, next one was Anthony van Leeuwenhoek. Okay, what was he working on? Ma'am, he was the person who worked on, he observed living cells. Okay, he observed living cells and named it little little animals. Okay, little animals. Why did he call it like this? Because they were moving rapidly. Okay, moving rapidly under the microscope. Okay, in the field of the microscope it was moving very rapidly. He worked on what, students? He worked on pond water to see bacteria, protozoans. Okay, blood to see RBC, plant, thin slice of plant, okay, to see the living cells. Okay, va? Understood? So he observed the living cells. Adukapram who came, students? Nama Robert Brown. Okay, adukapram who came? Robert Brown. Okay, Robert Brown what was he noticing? He noticed, or he discovered nucleus or nuclei. Okay, he discovered nucleus. Here also I'll change the color. Okay, yeah. He discovered nucleus in the cells. எதில் work பண்ணிட்டா? He worked on orchid roots. He worked on what? He worked on orchid roots. Okay va? This is understood? So this is the basic discovery. Idukapram we will be doing cell theory. Okay, after this we will be doing what? Cell theory. So this is your GM expression. Okay, done. Understood? Yes. So this is the whole thing. Now, students, one of the most important topics is your cell theory. Students have lot of confusion here, so we will go very slowly and try and understand everything, so you can watch it at your own pace. In 1838, okay, Matthias Schleden examined a large number of plants and observed that all plants, okay, Schleden one the students, he observed what? All plants are composed of different kinds of cells which form the tissues of the plant. So he is telling that ella plants there are different kind of plant cells which are present. Okay, this is what he was trying to say. All plants have cells. Okay, next person who came today, students, it was Schwann. All of you remember in neuron chapter there is Schwann cells which secrete myelin sheaths. So remember, Schwann cells or Schwann person is related to animals, okay, and Schleden is related to plants. So Schwann studied different types of animal cells and reported that, okay, so he worked on animal cells, and what did he report? That a thin outer layer which is today known as the plasma membrane is present, okay. This was the finding. What did they find out? He found out, okay, one person that is your Schleden. Okay, this is Schleden, this is Schwann. Just make sure that you know their faces as well. Okay, they will not ask, but then we can't predict. Schleden worked on plant cells. Okay, plants. He worked on different plants. அதுக்கப்புறம் என்ன நோட்டீஸ் பண்றா? அதுக்கப்புறம் he noticed that different kinds of cells, okay, which form the tissues are there. So he noticed that there are different kinds of cells, okay, different kinds of cells. This was his finding, very simple finding, that all plants have cells, okay, and he was, students, a German botanist. Okay, obviously a person who is working on plants should be a botanist, okay, German botanist. Correct தானே? Yes ma'am, correct. Okay, அதுக்கப்புறம் students Schwann. So Schwann cells you can remember. So அது வந்து easy ஆகும். Schwann cells you can remember. So Schwann was working on what? He noticed that, okay, he noticed that what? He was working on animal cell. Okay, he was working on animal cell, and he noticed that in the animal cell ஒரு thin layer இருக்கு. Okay, ஒரு thin layer இருக்கு, which is the plasma membrane. Okay, which is the plasma membrane. Okay va? Okay va? Understood this? A plasma membrane. Okay, very important it is. Make sure that you remember it. Okay, make sure that you remember it. So these were the two findings, but again it's not the end of it. So look here, Schleden and Schwann together formulated the cell theory. So what did they work on? They called something called as the cell theory. This theory, however, did not explain how a new cell is formed. So both of them were working. One was working on animal cell, one was working on plant cell. Ana and the theory they were not able to tell how a new cell is actually getting formed. So அது வந்து கொஞ்சம் டிஃப்ரெண்டா இருந்துச்சு. So we wanted one more person to join this cell theory that we will understand. Okay, now look here. All living organisms are composed of cells. This is the first finding. All living organisms. Each cell is made up of small mass of protoplasm which contains the nucleus inside it and a plasma membrane with or without a cell wall on its outside. Cell wall is there for the plant, right? So each cell, students, they have a small mass of protoplasm. Protoplasm and the fluid, cytoplasm plus nucleus, okay, which will have nucleus inside, and it also will have a plasma membrane with or without cell wall. Cell wall with or without cell wall, okay. Activities of an organism are the sum total of activities and interactions to its constituent cell, which we tell know metabolic, okay. Now Viruses are acellular and they do not have cellular machineries. So actually when these people are working, they notice that this virus fellow is not coming and falling anywhere. He is not falling in living also, he is not falling in non-living also. Now also the debate continues and the debate ku vandu answer kediyadu, because virus when it comes in contact with a host it will automatically become living. Ana without a host it will act like a non-living. So adanale virus are acellular and do not have a cellular machinery. Okay, this was the basic thing, students. But again the conversation comes that what about new cells? How are new cells actually formed? So look here, Rudolf Virchow, okay, in 1855 first explained that cells divide and new cells are formed from pre-existing cell. Okay, pre-existing cell. Okay, he told that you people are very confused, don't worry, but please understand that one cell, from one cell only another cell will be formed. So he told that first explain that cells divide and new cells are formed from pre-existing cells. Pre-existing cells divide and they form the new cell. He called this as omnis cellula e cellula. Okay, omnis cellula e cellula, ஒரு song மாதிரி omnis cellula e cellula, omnis cellula e cellula, அது மாதிரி ஒரு rap you have to make in your head so that you don't forget this term. Now students, imagine all these findings had to come together. All these findings had to come together because namakku vandu Schleiden had told that okay, all the plants have cells. Okay, Schwann had told that all animal cells have a thin outer layer which is called the plasma membrane. Ana idu rendu theory ilme oru point missing da, how a new cell is formed. Adu vandu Rudolf Virchow gave. So இந்த மூணுமே combine பண்ணி நம்ம modern cell theory form பண்ணலாமே. So here you can see he modified the hypothesis of Schleiden and Schwann to give the cell theory a final shape. Okay, that is all living organisms. Okay, what was it, students? All living organisms are composed of cells and product of cells. Okay, all cells arises from pre-existing cells. Make sense? Yes or no? Does it make sense? Yes ma'am, it makes sense. Correct. This is the modern cell theory. Modern cell theory. Okay. So, write a flow chart. Okay, look here. Okay, this we will come into. One second, students. Let's write it here. Okay, so here to find out that in a better way, an overview of cell also, you should remember that if I take an onion cell, okay, if I take a onion cell, you have done no experiment in your first PUC, I believe. You would have taken the onion cell and done an experiment where you removed out that white color layer and the onion cell remove பண்ணும்போது students, what do you observe? You observe a cell wall. Yes or no? You observe a cell wall. So, on the cell wall it is present in the plant cell. Okay. So, adinala it will give a definite shape to the plant cell. Yes or no? Yes ma'am, correct. So, look here. Okay. I will try to close this. It's like your brain, it's not loading. Okay, it's not loading. One second, students, it's getting stuck. It's telling me that it will work. Okay, so we can have hopes. Sometime it will hang, students, we can't do anything. Okay. Okay, let me try it after some time. It's working. Okay, it's working. Can you see? This is a plant cell. And the plant cell க்கு ஒரு definite shape இருக்கு. Okay, பார்க்கும்போதே you will understand. அதுக்கு வந்து ப்ராப்பரான ஒரு ஷேப் இருக்கு ஸ்டூடெண்ட்ஸ். அந்த ஷேப் வந்து it is because of the நம்ம cell wall தான். Okay, இந்த ஷேப் வந்து it is because of the cell wall. See how proper the shape is, right? So same thing, when you took that onion cell and you removed it out and you saw it under the microscope, ப்ராப்பரான ஒரு cell wall இருந்துச்சு. பட் அனிமல்ல அது கிடையாது. அனிமல்ல வந்து only plasma membrane is there, and this plasma membrane will help for what? The transport. Okay, substance எதெல்லாம் enter ஆகணும், எதெல்லாம் exit ஆகணும், அந்த ஒரு regulation இருக்கும். ஆனா இந்த cell wall வந்து it gives a definite shape and that rigidity to the plant cell. Okay வா? So this is the finding. Okay, onion cell cell wall. Then students, if you have to take the animal cell எப்படி ma'am extract பண்ணுவே? Students, you can take it from your cheek. Okay, now we have all done this. Your cheek cells. Okay, from the cheek. In the cheek illa students, cheek. You use a small like a scapula types, you scrape it out, you will get the cheek cells. And the cheek cells illa vanda, if you see it under the microscope, you will be able to see the animal cell. Okay, you will be able to see animal cell, and that is your plasma membrane. Okay, plasma membrane, and here right you will have a nucleus. So this is the finding that we saw, what these people are telling so far as well. Plasma membrane and center la vanda nucleus irukku. Okay, center la vanda nucleus irukku. So this is understood, right? This is the overview of cell. Remember, in the nucleus la vanda what will be there students? Nama nucleus la vanda genetic material irukum. So if you take a magnified image and look at the nucleus, you will start seeing the genetic material. At the close up la pathena, you will be able to see the chromosomes. Okay, in the chromosome la nama DNA genetic material is going to be present. Clear? Which is the genetic material. This is the basic. Okay, so this is the overview of cell. Okay, we'll put it in a box. Okay, we'll put it in a box. Oh, before I put it in a box, 1 second, we'll move it to one side so we can write the cell theory as well. Cell theory is very important. Okay, very very important. So miss panama, make sure that you write and keep it somewhere, because you will forget it otherwise. Okay, you will forget it otherwise. Okay, cell theory la main enna students, nama cell theory la, okay, cell theory first it was given by Cytoskeleton and Schwann. Okay, Cytoskeleton and Schwann. Cytoskeleton vanda, he told that all plants, okay, all plants will have cells. Okay, and it is made up of different types of cells which will combine to form tissues. This is the basic, so you can just write all plants have cell. Schwann vanda, he told that animal cells have plasma membrane. Okay, thin layer of PM. Okay, PM na plasma membrane. ஆனா இந்த செல் தியரி கம்ப்ளீட் பண்றதுக்கு we wanted one more person, which is Rudolf Virchow, who told us that cells come from pre-existing cells. He told what? Cells are formed from pre-existing cells. Okay va? So new cells are formed from pre-existing cell. So this was the thing. So இது எல்லாமே combine பண்ணி நம்ம வந்து ஒரு modern cell theory, okay, modern cell theory came into existence. Okay, modern cell theory came into existence, which spoke about all the points. Okay, all the 3 points. Okay, all the 3 points were combined here. Okay, அதுதான் இந்த point students. Okay, இதுதான் இந்த paragraph. All living organisms are composed of cell and products of cells. All cells arise from pre-existing cells. So இந்த point வந்து correct, it was told as the modern cell theory. 3 points combined panni, we came up with the modern cell theory. Okay. Now we are going to study certain size, shape of certain organisms. Okay, certain organisms. So, inga paakumbodhu students, see, இது வந்து NCERT-ல இருக்கிற diagram, so அதனால important தான். Cells are of various shape and size. Okay, mycoplasma, the smallest cell. All of you remember in biological classification we spoke about the cell which does not care if oxygen is there, which does not care about cell wall, which is one of the smallest living cell known. So and the mycoplasma da. So mycoplasma, the smallest cells are only 0.3 μm in length. Bacterial cells are around 3 to 5 μm. The largest isolated single cell is an egg of an ostrich. Okay, the largest isolated single cell. This is the largest. Okay, the largest cell is of an ostrich. Is of an ostrich. So red blood cells வந்து students round and biconcave இருக்கும். White blood cells வந்து amoeboid shapeல இருக்கும். Columnar cells long and narrow இருக்கும். Nerve cells வந்து branched and long இருக்கும். So long and large difference. Okay, and then students a tracheoid, elongated, mesophyll cells round and oval. Okay, so here you can see the human red blood cells are about 7.7 μm in diameter. Nerve cells are some of the longest cells. Longest na height adhigama irukum. Okay, long length. Cells also vary greatly in their shape. They may be disc-like, biconcave, polygonal, columnar, cuboidal, squamous, அது மாதிரி எவ்வளோ shapes இருக்கு. Okay, அவர் வந்து shapes எல்லாம் vary ஆகும். The prokaryotic cells are presented by bacteria, blue-green algae, mycoplasma and PPLO, which is pleuropneumonia-like organisms. So, the prokaryotic cells, primitive cells are represented by நம்ம mycoplasma, bacteria, PPLO in the cells எல்லாமே. Okay, one second, I'll drink some water. Okay, so students, I would say it's better to learn this entire thing in one box. Okay, so this we already know, no, in biological classification we have studied that nama bacteria vanda shape divide பண்ணும்போது 4 shapes we have, spherical, comma shaped, spiral as well as your vibrio shaped. Okay, so comma shape, sorry, spherical shape, rod shaped, comma shaped, spiral shape. So basis on the shape as well. Now here what you have to remember is and the numbers ellame important da, so and the numbers ellame correct da, you should remember. Okay, so in the box la edudala students. Okay, idile prokaryotic, eukaryotic try pannalam. Okay, inga vanda cells, okay, so இப்போமே எல்லா animal cell எல்லாமே we will write it at one point. Okay, smallest cell வந்து நம்ம mycoplasma, correct தானே? One more we will add. Okay, yes, this should do the job. Okay, done. Smallest cell வந்து students mycoplasma. What is the size of this students? Size வந்து எவ்வளோ இருக்கும்? 0.3. Very good. 0.3 μm. Okay, இவ்வளோ தான் size இருக்கும். So இங்க வந்து one second. Organism, அதுக்கப்புறம் size. Okay வா? This is all I'm writing, so your work becomes easier. My work is more actually, but your work should be easier for me. Okay, அதுக்கப்புறம் students next வந்து நமக்கு bacteria. Okay, இது வந்து prokaryotes, prokaryotic cell. Prokaryotic cell. Bacteria size வந்து எவ்வளோ ஆகும் students? Vary ஆகும் 3 to 5 mu m. ஆனாலும் இது வேரி ஆகிறதுக்கு சான்ஸ் அதிகமாக இருக்குது. ஸோ அதனால 3 to 5. ஓகே, ஆவரேஜ் வந்து ஓகே, திஸ் இஸ் ஒன் ஆவரேஜ் கேல்குலேஷன். அதுக்கப்புறம் நம்ம லார்ஜஸ்ட் செல். ஓகே, லார்ஜஸ்ட் செல் வந்து என்ன இருக்கும்? ஆஸ்ட்ரிச் எக். ஓகே, ஆஸ்ட்ரிச் எக். Okay, all of you tell me how much it will be. All of you tell me how much it will be ostrich egg. Approximate. Okay, you have to check and tell me. Yes? Some of you searched. What is it? Approximately, students, approximately 15 to 18. Okay, 15 to 18 and weight வந்து almost 1.5 kg இருக்கும். Okay, 1.5 kg இருக்கும். So, largest cell known, so 15 to 18 centimeter and kg வந்து 1.5 kg இருக்கும். அதுக்கப்புறம் ஓகே, அதுக்கப்புறம் largest cell ostrich egg understood. ஓகே, அதுக்கப்புறம் ஸ்டூடெண்ட்ஸ் நம்ம RBC. ஓகே, RBC வந்து how much? 7.0 mu m. இந்த shape வந்து it can be biconcave, it can be disc like shape, many different shapes of cells, shapes are there, no? Now நம்ம RBC வந்து somewhat like this it will be. Okay, like this it will be. Okay, அதுக்கப்புறம் நமக்கு வந்து columnar cells இருக்கு. இது மாதிரி long slender cells will be there. And in the columnar cells ஒரு main விஷயம் என்னன்னா நம்ம nuclei வந்து baseல present ஆயிருக்கும். Okay, are you all enjoying? Yes, are you all enjoying? The energy should keep going and share it with your friends as well. Okay, அதுக்கப்புறம் ஸ்டூடெண்ட்ஸ் நமக்கு வந்து cuboidal cells present ஆயிருக்கும். இது எல்லாமே ஷேப்பு தான். ஓகே, இது எல்லாமே டிஃப்ரெண்ட் ஷேப் அண்ட் சைஸு தான். இதில் வந்து நியூக்ளியஸ் சென்டரில் இருக்கும். ஓகே, அதுக்கப்புறம் நமக்கு ஸ்குவாமஸ் செல்ஸ் இருக்கு. எஸ் மேம், வி ஸ்டடிட் அபவுட் ஸ்குவாமஸ் செல்ஸ் as well. அந்த ஸ்குவாமஸ் செல்ஸில் என்ன இருக்கும் ஸ்டூடெண்ட்ஸ்? அந்த செல்ஸ் வந்து irregularly ஷேப்டாக இருக்கும். ஓகே, irregularly ஷேப்டாக இருக்கும். அதுலேயும் நியூக்ளியஸ் கண்டிப்பாக இருக்கும். ஓகே, இது மாதிரி எவ்வளோ செல்ஸ் இருக்குது நம்ம பாக்டீரியாவில் 4 shapes of cells இருக்குது. ஸோ இது எல்லாமே இம்பார்ட்டன்ட் தான். ஓகே, ஸோ RBC we understood, biconcave in shape. Then which is the longest cell? Okay, longest cell is going to be a nerve cell. Okay, longest cell வந்து நம்ம nerve cell தான். Okay, longest cell வந்து நம்ம nerve cell தான். Okay, understood this much? Yes ma'am, understood. Understood. Clear? Yes, you can write that also. Prokaryotic cells la 4 shapes irukka, adum you can write it, but Anna, you can just write this much only. This is very very important. Okay, clear? Okay, this is done. Then prokaryotes la vandu bacteria irukku, PPLO irukku, mycoplasma irukku. Okay, oh, longest cell approximately 1 meter long irukum students. Very long, no? 1 meter long. நம்ம scale வந்து 50 centimeter. 1 meter long approximate. Okay, இந்த values எல்லாமே approximate தான். So this is understood. இன்னொரு ஸ்லைடில் வந்து நம்ம prokaryotic cell எழுதலாம். That is, then prokaryotic cell will feel bad. Then you will tell me, ma'am, why did you do what is it? Partiality. Online teachers வந்து partiality we will show for students who attend regularly. Okay, because our name வந்து it will be in the mind. Okay, but in school and all, remember, partiality used to be for the topper students. Okay, who used to answer question properly. The topper student used to get so much attention from the teacher. Every time they used to be the class leader. Okay, that time how much jealous we all used to be, students. I have been class leader multiple times. Okay, so blue green algae. Okay, அதுக்கப்புறம் mycoplasma. But online students la enna, these won't be our students. Okay, like whoever is our regular student also, they go to every channel. So அதனால நமக்கு வந்து அந்த my student அந்த feeling ரொம்ப கம்மி. But ஆனா we will feel, teachers will feel. students will not feel like that. Oh, my teacher, she is my teacher, I should always be present in her class. அந்த ஃபீலிங் வந்து கம்மியாக இருக்கும். பட் ஆனால் டீச்சர்ஸ்க்கு வந்து ஓ தி ஸ்டூடெண்ட் இஸ் மை ஸ்டூடெண்ட், இந்த ஸ்டூடெண்ட் வந்து இந்த சேனலில் இருக்கிறாங்க. வென் வி see the chats we'll see the name and we'll be like ஐயோ the student went there also. ஸோ அந்த மாதிரி ஒரு ஃபீலிங் இருக்கும். ஓகே, ஸோ பாக்டீரியா, ப்ளூ கிரீன் algae, mycoplasma and PPLO. okay, pleuropneumonial cells. This is clear? Yes. Okay, now students, in the bacteria in the continuation அப்படியே எழுதலாம். okay, bacteria 4 shapes will be there which you have already studied in biological classification. ஆனாலும் it is important. Coccus, okay, Bacillus, Vibrio and Spiral. okay, so இதுதான் based on the shape and size. okay, this much only you have to study in prokaryotes. So this is done and dusted. okay, let's go to the next topic. So prokaryotic cell. So இதுதான் நம்ம prokaryotic cell. First you have to understand what is prokaryotic cell. அதுக்கப்புறம் we'll come to the shape and size and everything. okay, genetic materials lies freely in the cytoplasm which is called as a nucleoid. First of all students, நம்ம prokaryote eukaryote partitioning பண்ணும்போதே first dialogue என்ன மேம், prokaryotic cellsல ப்ராப்பரான நியூக்ளியர் மெம்பரேன் கிடையாது மேம், அந்த நியூக்ளியர் மெட்டீரியல் வந்து நார்மலா செல்ல இருக்கும், சைட்டோபிளாசம்ல இருக்கும். அதுதான் நம்ம ஃபர்ஸ்ட் பார்ட்டிஷனிங் ஆர் கிளாசிஃபிகேஷன். ஸோ அதுதான் நம்ம ஃபர்ஸ்ட் லைன். ஆல்சோ prokaryotic cell genetic material will lie in the cytoplasm freely. okay, it does not have a proper nuclear membrane. okay, peptidoglycan forms the structural network of the cell wall. இது வந்து நம்ம bacteria biological classification under Monera we studied that the cell, okay, the cell is made up of peptidoglycan, or else it was also called as what? Polysaccharide, amino acids plus polysaccharide, or else I told you one more word, amino sugars. அதுதான் நம்ம murin peptidoglycan சொல்லுவாங்க. So peptidoglycan forms the cell wall. okay, this we have studied in biological classification. Yes ma'am, we have studied this in biological classification under kingdom. This is how you should connect all your chapters together. okay, kingdom Monera we have studied polysaccharide. okay, polysaccharide plus amino acids, correct? yes, together what can you call it? You can call it peptidoglycan. இல்லைனா நமக்கு வந்து murin கூப்பிடலாம், MUREN. okay, murin. clear? This is the, I'm writing it there because 11th standard students they will not know. okay, peptidoglycan forms a structural network. Peptidoglycan has repeating units of, this I had taught you before and so many students are like ma'am, why are you teaching so much additional? see, cell chapter la verum indu. okay, NAG and NAM students. okay, repeating unit of enna irukum? NAG and NAM. okay, NAG என்ன? N-acetyl glucosamine. NAM என்ன? N-acetyl muramic acid. So in the combination, in the repeating combination that forms the peptidoglycan structure. 70S ribosomes are present. okay, 70S ribosomes are present. Mesosomes are folded invaginations in the plasma membrane of the bacteria, naked. In the line வந்து அப்புறமா பண்ணலாம். okay, I'll teach you in detail that. Naked circular DNA known as plasmids are present. okay, this also we have studied biotechnology chapter which I'll be doing, which I've done. So on the chapter students we studied that நம்ம cells normally நம்ம cells draw பண்ணும்போது we know that we have one set of genetic material. okay, இது மாதிரி genetic material இருக்கும். other than that நமக்கு வந்து ஒரு extra chromosomal circular DNA இருக்கும். அந்த extra chromosomal circular DNA வந்து என்ன இருக்கும்? plasmid-ஆ இருக்கும். அந்த plasmid வந்து நம்ம எதுக்கு யூஸ் பண்றா? அந்த plasmid வந்து we are using it as a vector. vector-னா என்ன students? vehicle. vector-னா என்ன? it is a vehicle. vehicle spelling. okay, when SIC and all comes is too difficult to understand. okay, we are using this plasmid as what? We are using this plasmid as a vehicle. okay, Understood this much? Okay, we have to study detail of this, don't worry, we have to study detail of this. So, right now we have only studied that outside one structure is there, and the structure is made up of peptidoglycan, which is made up of repeating units of NAG and NAM. Okay, N-acetyl-muramic acid, N-acetyl-glucosamine, 70S ribosomes are there, and they have a naked circular extra chromosomal DNA which is called plasmid, which acts as a vector, which we study in biotechnology chapter. Okay, now animal cell, plant cell. Okay, see here, prokaryotic cells are primitive cells. Okay, primitive cells that are found in bacteria, mycoplasma, etc. Avarku vandu organelles kediyadu students. No organelles like the ones in eukaryotes are found in prokaryotes except for ribosome. Ribosome vandu a universal organelle. Ella prokaryotes, eukaryotes ellathume it is present. So we call this a universal organelle. Okay, we call it a universal organelle. Okay, prokaryotes are something unique in the form of inclusions. Okay, in the inclusions vandu romba important. Avarku vandu some foldings are there which are called as mesosomes. Other than that, they have certain regions which are called as inclusions, and the inclusions la vandu students, there are so many things that can be present. Okay, like chromatophore, like pigments and other things, which I will teach you again in detail. So a specialized differentiated form of cell membrane called mesosome is characteristic of prokaryotes. They are essential infoldings. Okay, nama cell la vandu idu madri infoldings will be present. Okay, like this infoldings will be present in the cell, which are actually called the mesosomes. Okay, which are actually called what students? Mesosomes. Okay, lot of functions we will have. Okay, replication, other things, they will be helping. Okay, continue பண்ணலாமா? So prokaryotics have something unique in the form of inclusion bodies, and the inclusion bodies can be phosphate storage, glycogen storage, and the madri specialized bodies will be there. Other than that, cell membrane la vandu specialized structures are going to be there. A specialized differentiated form of cell membrane called as mesosome is a characteristic of prokaryotes. They are essentially infoldings of the cell membrane. So let's just draw this and understand, students. So let's first draw a very, very simple cell. Okay, you just draw a very simple cell like this. Yes ma'am, this is a very simple cell. Then we have to draw inside. Okay, this is the cell membrane, and the cell membrane, students, idu vandu foldings, like infoldings it will be formed. So here you can see tubular infoldings, you can see lamellar infoldings. Okay, or you can see vesicle infoldings. Idellame different type of mesosome foldings da. Okay, all these are different types of mesosome foldings. Other than that, generally in smaller classes also we have studied that they will have a nucleoid. Okay, where the genetic material is there. Okay, they are going to have that, and they are going to have plasmid, which is a circular, okay, double stranded circular small extra chromosomal structure. Okay, this much we know. Then we know these people are going to have ribosome and the universal organelle. Just now I taught you that. So they will have this 70S ribosome. Okay, then they spoke about one set of group of proteins which are there, or different kind of bodies which are there. They are called as your inclusion bodies, which are going to be like this. Okay, they are like just an aggregation type. In the madri present aayirukkum, there will be different inclusion body. One will be phosphate, one will be glycogen storing. Okay, in the madri irukum. Okay, like this inclusions bodies will be present. Okay, adukapram what will be there, students? Other than that, normally what will be there? Here comes your one very important thing, that is going to be your flagella. Okay, your flagella, which is going to come from your cell membrane. So it will extend like this. Okay, it will extend like this and it will come. This is the flagella which will be formed. Like this, fimbriae will be there, pili will be there. Okay, so all this will be present. Then we also have one more thing which is called as your chromatophore. Idu vandu enna pandranga? Pigment. Okay, it is for the pigment. So again we will draw it here. It extending like this inwards, pointed, and like this, where they have pigments. So this is the structure. I'll explain everything. Don't have to worry. Every single thing I'll explain it properly for all of you. Just make sure that you're understanding so much. Okay. You're understanding this much, right? Yes. Okay. Now let's start labeling each of it. Now what and all we know, students, we have outer layer, which is going to be the cell wall. Okay. Glycocalyx, we will write all that later. Okay. This is going to be your cell membrane. In the cell membrane, some type of infoldings are going to be present. In the infoldings, which is called as mesosomes. Mesosomes. Okay. This one the tubuli. Okay. Tube shape madri irukku. So another tubuli. This will be your lamella. And this is going to be vesicles. Okay, vesicles. So these are the different shapes and types of mesosomes that you can see. In the mesosome one, the students, it helps in respiration, it helps in cell wall formation, and the madri it is taking care of majority of the functions, like it is helping in respiration. Okay, it is helping in cell wall formation. I'll show you from NCERT, don't worry. Okay, cell wall formation. This is understood. Now we will go to the next labeling. This is what we have. Our genetic material is going to be here. Genetic material is going to be present here. Correct, ma'am. This is what our DS okay. I'll just write plasmid. In the plasmid one, it is extra chromosomal. Extra chromosomal structure, right? Extra chromosomal structure. So that is your plasmid. Correct, ma'am. Understood? Circular, yes. Naked, we can call this the naked DNA, or we can also call it like a nucleoid. This also you people are already aware. This one the number nucleoid. Okay. This now coming to this, students. This one the number inclusion bodies. Another inclusions bodies. In the bodies one the special in prokaryotes. So in the inclusion bodies one they can be used for, let's say, phosphate storage. They can also do the glycogen. So on the madhi multiple functions will be taken care by your inclusion bodies. Okay. So we will write some of them. Store glycogen. Okay, store glycogen. They can also be phosphate bodies. Okay, phosphate bodies. They can do this. Now we say gas vacuole. Have you heard this word, gas vacuole? Okay, gas vacuole. So in example here is gas vacuole. We will read NCERT for you to have better clarity as well. Okay. Gas vacuum. Then students, if you look here, you have something that is breaking open or just say moving out of the cell wall and emerging from the cell membrane. So this is your flagella, which is going to help in what? This is going to help in movement. Okay, this is going to help in movement. Okay, movement. So flagella will open out, that is, it is emerging from the cell membrane, okay, and it is moving out of who? The cell wall, right? It is coming like this. So this is the flagella structure, and this is what you are seeing here. This is your 70S ribosome, because I told you ribosome is universal, so it is going to be present no matter if it is pro or U, it is going to be present. Okay, so this is the basic structure. I hope this structure is clear for all of you. Clear? We will learn more about it, don't worry. But then right now you have to understand this much. Again, I'll teach you about this as well. And here students, here comes a very important thing which is called as your chromatophore. Chromatophore. From here, chromatophore word only you will understand that pigments, right? These are going to have some pigments, right? Chromatophores, pigments. So this is the cell which will have whatever is important for us to study so far, everything is there in this diagram. Okay, now we will study detailedly about each of it, but first you should understand this diagram properly. Okay, understood this? So students, first namakku vandu oru cell wall irukku, adukapra oru cell membrane irukku. Remember I showed you 3D also sometime back. So oru cell wall irukku, adukapra oru cell membrane irukku. From the cell membrane you will see emerging flagella which is there, which is going to help with motility. Okay, adukapra nama cell membrane there some infoldings are present which are called as mesosome. They are going to help us with respiration, cell wall formation, and the madri important ana vela ella in the mesosomes are going to help us with. Adukapra namakku vandu plasmid irukku, which is an extra chromosomal circular DNA structure. Adukapram genetic material or your naked DNA, because prokaryotic cell will not have a proper nuclear membrane, so adanale oru nucleoid structure irukku. Then we have something called as inclusion bodies. Inclusion bodies na or aggregation of proteins which are going to help in storage of glycogen, phosphate, and example vandu nama gas vacuole. Okay, then we have a ribosome which is your 70S ribosome. Idudha oru general structure. The structure I have drawn just so you people can understand. Okay, now look here. Mostly eukaryotic cell, particularly the bacterial cell, have a chemically complex cell envelope. Okay, cell envelope. The cell envelope consists of tightly bound 3-layer structure. So students, enna solranga, they are saying that this prokaryotic cell or your cells itself, what they will have 3 layers. That is, your first layer is going to be a glycocalyx layer which is going to do the protection. Okay, all the 3 are actually going to act as a single envelope and do the protection. Ana first nama vandu outermost layer called glycocalyx, which is followed by nama rigid layer which is called the cell wall. Okay, nama rigid layer which is called the cell wall. Adukapram then we have our plasma membrane which is selectively permeable. So adukapram nama plasma membrane. So in the madri 3 layers will be present. Okay, in the madri 3 layers will be present. So as I am reading the NCERT lines, idellame NCERT lines da. So as I am reading the NCERT lines, I will be writing the notes in the side itself, so you people don't have to, you know, look elsewhere. Okay, let me just wear my glasses. Okay. Okay, yes. So students, nama first layer vandu enna ma'am, first layer vandu nama glycocalyx. Okay, glycocalyx na enna, sugar layer. Okay, that means what? Carbohydrate. So your glycocalyx means what? Basically a carbohydrate layer. Carbohydrate layer. In the glycocalyx on the mainly help agum students, and the glycocalyx is mainly going to help this bacteria, right? All this prokaryotic cell vandu nama bacteria. Ipo in the penilla bacteria irukku, on my hand there are bacteria, everywhere in my body there is bacteria, everywhere in your body there is bacteria. Ana this bacteria has to get protected when it enters into the host organism. It has to be protected. So for protection, this glycocalyx is going to help. So it will protect it from the host, but again, it is there to protect. That doesn't mean any bacteria enters anywhere, it will be protected. Nothing like that. There is no assurity like that. They will try to protect, but confirmation of protection if they will die or not, that is only, you know, up to the host, how strong the host immune system is. But glycocalyx job is going to be protect from the host. Okay, protect from the host. So that is what it is going to be. Second one, the nama cell wall. Okay, second layer vandu nama cell wall. In the cell wall vandu mainly enna pandranga students, cell wall vandu rigidity. So it is helping to the cell to be rigid and in shape. Okay, and for the shape it is going to help. That is the function of your cell wall. Okay, adukapra nama last layer da nama plasma membrane illana nama cell membrane nu koopuduvanga. So in the plasma membrane vandu selectively permeable ah irukum. Okay, who should enter, what should enter when the plasma membrane will decide. Okay, it will not allow anyone else to decide. So plasma membrane is selectively permeable. Selectively permeable. Just like your mother. Your mother will not like all the friends of yours. Your mother will like that one friend who is the most enna solranga, most rowdy madri irukra friend, your mom will be like, oh, he's so nice, she's so nice. Ana good friend she'll tell avoid, okay, and she will like this friend because this friend is very good to pretend. So அந்த மாதிரி we will all have one best friend who is very very, you know, we know that this person is the worst person that I shouldn't be friends with, but your mother will tell he is a very good friend of yours, be with him. So this is the 3 layers, students. Although in the glycocalyx, understand நம்ம வந்து separate we are learning about it. Ana kuda students, in the glycocalyx mattapadi nama cell wall, cell membrane எல்லாமே ஒரு single envelope மாதிரி தான் function பண்றாங்க. Okay, it is working as a single unit, and that single unit is going to do what? Protection of the cell. Protection of the cell. Okay, understood this? Protection of the cell. Correct, no? We will learn more about it. Okay, we will learn more about it, don't worry. But Anna, this is important for you to remember. Okay, done? Yes. So although each layer of the envelope performs distinct function, they act together as a single protective unit. Students, in the cell chapter without nama NCERT, it is very difficult to, you know, make you also understand, and it is very difficult for you to just understand when I draw. So adinala da I'm keeping NCERT and drawing side by side so you understand. Okay, later we will also do some revision notes. Anna, this is the best method you can, you know, we can teach you. This is the best method. If you write the whole thing also, very few students will understand then after reading NCERT. So adinala NCERT lines plus this will help you a lot. Okay, understood this? Okay, now we will learn more about it. Okay, mesosome function inge irukku, students, whichever I told you, no, in the mesosome function inge irukku. Mesosomes are in the form of vesicles, tubules and lamelles. I drew that only for you. This is your tubule மாதிரி இருக்கிற structure. அதுக்கப்புறம் lamel மாதிரி இருக்கிற structure இந்த மாதிரி இருக்கும். Okay, இந்த மாதிரி இருக்கும். And அதுக்கப்புறம் vesicle மாதிரி இருக்கிற structure. Okay, இதுதான் நம்ம mesosome. So they are tubule, okay, vesicles, so vesicles, tubules and lamellae. Okay, I didn't draw it in order. They help in cell wall formation, DNA replication and distribution to the daughter cells. Distribution to the daughter cells means the daughter cells will be like this. These people are going to catch the head, like how you will catch the head, no, and push like that. And the madri, they will catch like this and they'll push it to both the sides. So adanale they will help in the distribution of the daughter cells. They will help in the cell wall formation. Adukapram namma DNA replication. They also help in respiration, secretion process, and to increase the surface area of the plasma membrane and enzymatic, and any infoldings for that matter, they will automatically increase the surface area. There's nothing to be specially told about it. You know that it will automatically increase the surface area. Okay, now let's learn more about nama glycocalyx. Okay, glycocalyx differs in composition and thickness among different bacteria. It could be loose sheath called as slime layer. So students, nama glycocalyx la 2 types of classification irukku. Okay, 2 types, like the cell envelope can be of 2 types. Okay, what is the 2 types? First one, the number tough capsule. In some bacteria, this glycocalyx will be a tough capsule. Okay, tough capsule, that means just like how I drew, no, that time, just like that, it will be like a proper tough capsule. In the madri irukum. Okay, very strict and very tough. Okay, in a cell bacteria, it will be like slime layers. Okay, slime layers, that means they are going to be present something like this. Okay, like this loose, like this will be slime layers will be present. Why so? Namakku vanda slime na it will have the water. Okay, and it is very easy to, you know, just insert it anywhere. Like example, நம்ம car clean பண்ணும்போது நம்ம வந்து brush use பண்றாங்க, but ஆனா with the brush the cleaning will not happen properly. So what we will use slime, and we will just keep it in the cup holder and everything, and we will clean all the dust. So அந்த மாதிரி தான். So here you can see this cell envelope will be 2 types. First one is going to be your slime layer. Okay, slime layer இருக்கும் in some, while in others it may be thick and tough called as the capsule. So that is why I told you tough capsule. The cell wall determines the shape of the cell and provides a strong, okay, already functions we studied. Cell wall will determine the shape of the cell and will provide a strong structural framework, just like our skeletal system. Nama skeletal system eppadi namakku andha support, this framework, this body, everything straight it is standing because of the skeletal. Andha madri da in the cell wall also will provide the rigidity, structure and shape for the cell. Okay, so the cell wall will determine the shape of the cell and provide strong structural framework to prevent the bacterium from bursting or collapsing. Now the plasma membrane is selectively permeable. This we already understood, that the plasma membrane is selectively permeable in nature and interacts with the outside world. So it does what? Interaction with the outside world, right? This membrane is similar structurally to that of eukaryotes. Is this clear to all of you? So இதுதான் நம்ம slime layer, இதுதான் நம்ம capsule layer. நம்ம வந்து streptococcus pneumoniae in Griffith's experiment பார்க்கும்போது நம்ம வந்து rough capsule, rough strains, smooth strain, அந்த smooth strainல வந்து ஒரு virulent ஒரு capsule layer இருந்துச்சு, rough strainல அது இருக்காது. So அந்த மாதிரி தான் slime layer and your capsule layer. Okay, so again lines of NCERT only. Okay, lines of NCERT only. Done? Understood? Okay. Okay. Now, okay, again they are explaining about mesosome a little. We have already studied, so we can get past this. So this is the slide, that one paragraph I showed you, no? Instead of that, this one paragraph is there, that there is a special membranous structure in the cell which is called a mesosome, which is formed by the extension of the plasma membrane. Extension of the plasma membrane. Main function in students helps in cell wall formation, okay, adukapram DNA replication and also distribution of the daughter cell. With that they also help in respiration, also secretion, as well as increasing the surface area. Okay, same thing they have given it here. I'll just push it here only. Okay, yeah. Next comes inclusion bodies. Okay, inclusion bodies na enna? Reserve material in the prokaryotic cell and are stored in the cytoplasm in the form of inclusion bodies. These are not bound to any membrane system and they lie freely in the cytoplasm. Example, you will see phosphate granules, you will see cyanophycin granules, that is your pigmentation. Illena, you can see glycogen granules. So the granules means what? Like the structures which you are seeing, one of it can be a phosphate granule. Okay, let's assume this is a PO4 3- granules, that is phosphate granules. Adukapram you will see one more granules. They are all free in the cytoplasm. Okay, இந்த மாதிரி ஃப்ரீயா இருக்கும். Like this, they will be free in the cytoplasm. They are not attached to any membrane. Let's say this will be your glycogen granules. Okay, glycogen granules. Are you all smashing the like button? Quickly go and mark your attendance. How do you mark your attendance? By smashing the like button. Okay. Gas vacuoles are found in blue, green algae and purple and green photosynthetic bacteria. So that is also an example of this, your gas vacuoles. Okay, understood? So very simple. Mesosome we have studied, inclusion bodies we have studied. In the inclusion bodies, they are basically reserve materials. They will have certain reserve materials. They are reserve material in prokaryotic cell which are in the cytoplasm. They are not attached to any membranes. They are not attached to any other organelles. They are free, happy people. Okay, free people. Phosphate, they can be granules, glycogen granules will be there, cyanophycin granules will be there. It ellame will be present in the cytoplasm. Okay, okay. Now we will study about our most important flagella. Okay, so students, nama flagella draw பண்ணும்போது I want all of you to remember your hair particle. Okay, so this is the flagella, right? Now you just imagine your hair that you have seen in your ad and everything. Nama Sunsilk ad illa ellame they show the hair from the root. Okay, imagine your hair was attached without a proper, like, you know, root was not there. Normally we see our hair like this. Okay, this is our hair. Ana downwards, in the hair downwards there will be a big root which is attaching this hair, correct? Yes or no? Like this one root will be there. Adinalada our hair will not just come out like that. Okay, otherwise every time you bath you will have become motte. Okay, so that's why I'm telling you, you should remember your flagella just like that. So in the flagella, students, same in the madri da, there is a basal body. Okay, there is a basal body. In the madri or basal body will be present which is helping in the attachment, and then also in the right in the center here they will have one more structure which is called the hook. again helping them to hold it together. So this structure is called the basal body, this is called the hook, and this is called the filament. So nama flagella mainly 3 parts are going to be present. Idu vanda nama filament, this is the filament, okay. This is the basal body. இதுதான் நம்ம hook. Okay, this is the hook. So the filament is going to be attached with the help of a basal body, and from there your filament is going to arise, which is the longest part of the flagella. Bacterial cell may be motile or non-motile. If motile, they will have thin filamentous extensions from their cell wall, which are called as flagella. What they will have? Extensions from their cell wall. Now here students, see, there's a confusing part here. They're saying extension from the cell wall. Actually the cell membrane, from the cell membrane only this emerging is happening, but they are extending from where? They are extending from the cell wall like this. Okay, this is the cell wall. So they will be extending out of the cell wall. Bacteria shows a range in number and arrangement of flagella. Bacterial flagellum is composed of 3 parts. What are they? Filament irukku, hook is there, and basal body is there. Okay, hook is there and basal body is there. The filament is the longest portion and extends from the cell surface to the outside. Cell surface to the outside. Is this clear? Yes, clear? Okay, so filament is there. I don't know why the color is very dull, students. I think I'll just make myself more dark. Okay, now it should be better. Okay, yeah, I'll be a little dark, but this will be visible better. So this is understood, students, nama filament. Okay, these all, this filament, pili, okay, sex pili, we say sex pili, and your fimbriae ellame surface bodies. They are in the surface. Okay, they are surface bodies. So I'll write a tabular column after that. So look here, beside flagella there is pili and fimbriae that are also surface structures of the bacteria, but they do not play a role in motility. So many students get confused. Nama vanda cilia padikkumbodhu we talk about motility, flagella padikkumbodhu we talk about motility. Ana pili and fimbriae patti pesumbodhu motility patti pesada, because they are not helping in motility. Motility na enna? Movement. They are not helping in movement. Okay, the pili are elongated structure, tubular structures made of special protein structures. So in this pili is actually famous when we talk about conjugation. Okay, when we talk about conjugation, idoru cell, okay, this is another cell. Between this, if there has to be a exchange, okay, that is sexual exchange has to happen, that will happen with the help of a very small tubular structure. This is called as your sex pili. Okay, sex pili, we usually learn it in conjugation, and you people don't have it, so adanale you don't know. So the pili are elongated tubular structures made of special proteins. Now the fimbriae, which is the special protein, pili will be made up of pil-in protein. Okay, the fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help them attach to the bacteria, to the rocks in the stream, and also to the host tissue. So this fimbriae's main job, students, is going to be to help them with the attachments. Okay, help them with the attachments. Clear? Let's just write that here. Okay. Okay, now students, we are studying about surface structures. Okay, enalla surface structures are present nama body la, like in prokaryotes, surface structures. Okay, first you have your flagella. Okay, first nama diagram draw panla, adukapram nama vanda explanation we will write. Okay, this is the flagella. This flagella will have a basal body, which we can draw it like this also. Okay, like this also we can draw, like a motor type. Okay, and then we will have a hook right here. Okay, a hook right here. So this is the flagella. Then we will draw the Pili, actually I told you, no, the sex pili is more easier to be understood because sex pili is what you people have not studied it, but just understand there is this structure. So here I will just draw two cells. Okay, two cells. We say positive sex pili, negative sex pili. Okay, this is the tubular structure through which the conjugation can happen. So that brings us to the second one. Okay, and the last one is going to be never a straight line. Okay, is going to be the fimbriae. So this is our cell. Okay, on the cell we will see some structures like this. These are your fimbriae. Okay, these are your fimbriae. So structure is clear for all of you. Now we will write the explanation for it. So first one vandu nama flagella. So idudha nama flagella uda filament, which is the longest part. Okay. Okay, longest part. Idu vandu hook. Okay, idu vandu hook. Idu vandu basal body. Okay va? Basal body. In the flagella main function enna students? Ma'am, obviously ma'am, flagella will do, if the bacteria is motile, then it is going to help in motility. Okay, idu vandu it is going to help in motility. Adha main job. Okay, flagella main job vandu motility da. Avlodha main job. Okay, then we don't have to worry about anything else. Now pili vandu, this is the pili. Okay, sex pili we can say. So this is going to actually help in conjugation. Okay, conjugation. So that is the main job of your pili. As such, there is no function. Here also you can see they have just written that they are elongated tubular structures made up of special proteins. Ingayum function kedayada, but Anna, here I'll tell you, they are made up of special proteins. Okay, made up of special protein. Enna and the protein? Pillin protein. Okay. Okay, protein. Clear? So it is going to be made up of it. Again, help in conjugation. Adhikapra students, namakku vandu small brittle-like structures. It mainly vandu students, this is helping with attachment. Okay, mainly in the structure vandu, it is helping with attachment. Helping attach where? Helping with attachment. Attachment to where? Ma'am, attachment to maybe a rock. Okay, to a rock or to the host body. So this is what this fellow is going to do. Clear? Small bristle-like structures help with attachment to the rock or to the host body. So these are the surface structures. Clear? So Gopika Mam notes is also ready, and I hope this is done. Done. Any doubts here? Please let me know in the comment section so I will answer all your doubts. Okay? Yes. Yes, one more thing I'll tell here. Now only I'll tell, later then it will become difficult. So students, namakku vandu in the flagella formation vandu, it is again done by certain proteins. In the proteins what is so important is that prokaryotes konjam different ana proteins irukum, eukaryotes in the flagella formation vandu different protein la irukum. So adanal later in the class you will be coming across a word called as microtubules. So there I will tell you that okay, this is going to help with motility. That time you should automatically understand, okay, tubulin protein is helping for the flagella formation in eukaryotes. Okay, so that is why this is important for you. I'll write it here only so that later you are not confused. So in prokaryotes okay, additional, not additional information, necessary information. Okay, in prokaryotes, it's your flagellin protein, which is helping for this, flagella formation. Ana eukaryotes, in eukaryotes, it is going to be your tubulin protein. What is it? Tubulin protein. Okay, so prokaryotes flagellin protein, eukaryotes tubulin protein. Here I am writing in bracket microtubules. So when I am teaching you this, you should be automatically understanding this microtubules are going to help in the flagella formation. Okay, done? Yes or no? Okay, this is done. One second, students. I have to teach you one more topic. The order in your NCERT and the order that every teacher teaches is completely different because in the chapter, order is very difficult to find. So, adanale we teach in different orders. Okay, now, animal cell and plant cell, nama general classification to understand so that when I get into eukaryote cell, it becomes easier for you. Animal cells are generally smaller in size. Okay, animal cells வந்து ஸ்மாலராக இருக்கும், பிளான்ட் செல்ஸ் வந்து இன்னும் கூட லார்ஜர் செல்ஸாக இருக்கும். செல் வால் வந்து அனிமல் செல்லில் ஆப்சண்டாக இருக்கும், அதனால தான் வி ட்ரா செல் லைக் திஸ். ஓகே, நம்ம வந்து இது மாதிரி செல் ட்ரா பண்ணுறா, ஆனால் இங்கே வந்து செல் வால் ப்ரெசென்டாக இருக்கும். ஸோ அந்த ரிஜிட் ஸ்ட்ரக்சர்னால we draw இந்த மாதிரி structure. Okay, then students, plastids are absent, இங்க வந்து plastids are present. Okay, then vacuoles in animal cell are many but they are small and they are temporary. Okay, temporary இருக்கும் நம்ம vacuoles வந்து animal cell but ஆனால் இங்க வந்து students they will be permanent and they will be very very large. Okay, large, they will be large central sap vacuoles. Animal cells generally have a single highly complex and a prominent Golgi apparatus. So they will have highly complex and prominent Golgi apparatus. Plant cell will have many simpler units of Golgi apparatus called as dictosomes. So this you don't have to know, but Anna, just know that they will have multiple smaller structures which are called as, again later we will study about Golgi apparatus. Animal cells will have centrosomes and centrioles, and a plant cell will lack centrosomes and centriole. This is important, this question has come in the previous year. Okay, so this one box is enough for you to understand the entire difference between your plant cell and your animal cell. I hope so far everything is beautiful. Okay, yes, beautiful. Okay, now I will write one more slide. Okay, one more slide about your cell wall. Okay, cell wall, like this only I will write. One second, students. Yes, we will write it here. Okay, we will learn all of this together. Okay, now I taught you about the cell wall. So this is going to be like a quick recap. Okay, quick recap. So look here, students. Namakku vanda there is a cell envelope. Okay, or a single unit of cell. In the cell envelope la namakku vandu starting la oru glycocalyx present aayirukkum. Yes or no? Yes ma'am, one glycocalyx was present. Okay, here additionally some things we have to learn, that is why I am writing it again separately. Okay, that is a glycocalyx. Adukapram namakku vandu oru cell wall irukkum which is rigid. Okay, adukapram namakku vandu oru cell membrane irukkum which is going to be permeable. These are our layers. Okay, now we have already understood what these layers are going to do. Anna, it is important for you to understand that we have studied nama chapters like in Monera and all. When we were studying, we have studied about pseudomurine, peptidoglycan. So like that we'll have to study a little more here. So this is your glycocalyx. Okay, this is your cell wall. Okay, this is your cell membrane. Okay, cell membrane. Now in these three layers are going to form a single unit. Okay, these three layers are going to form a single unit that is helping in protection, which is going to help with protection. Yes or no? Yes ma'am, it is going to help with protection. 100% is going to help with protection. Yes or no? Yes. Okay, now students, this glycocalyx vanda, okay, we are just taking it here. Okay, in the glycocalyx vanda already you can understand glycocalyx now. Okay, this is sugar something. Okay, so carbohydrate I can say. I can also say this is going to have a capsule like covering. So sometimes you can also say mucopolysaccharide. Mucopolysaccharide I can say. Yes ma'am, you can say. Okay, adukapram I told you in the glycocalyx in the cell envelope it can form ஒரு tough capsule, ஒரு tough capsule layer, or it can form what? A slime layer. Okay, slime layer. In the tough capsule layer வந்து students tough இருக்கும். Wow ma'am, mesmerizing answer. Okay, tough capsule layer வந்து students mainly it is going to help for protection. Mainly வந்து it is going to help for protection. Correct? Yes ma'am, it's going to protect because tough capsule then students, so adinale it should do protection. Yes ma'am. Okay, then we have your slime layer. In the slime layer வந்து more of like very slimy layer da. This one will actually do, it will prevent water or ions to be lost from the bacteria. Lost from bacteria. Okay, so this is the job of these people. Tough capsule irukku, adukapram slime layer is there. Okay, slime layer is there. Is this clear? Yes. Okay, namakku vanda next topic vanda nama cell wall. Okay, again gram positive, gram negative also we can learn here. That is students, gram positive na the one that will take up the stain, and when they take up the stain it goes through the layers and the entire cell is going to be stained and it will not remove that color immediately. Ana gram negative na it will just be present on the surface, so by the time I put the next color, that color will move and the next color will take up, so it is like a temporary relationship. Okay, ana gram positive and a permanent relationship madri. We usually use crystal violet color to check even bacillus, okay, shape, rod shape bacteria. Majority we do gram positive and gram negative. Okay, namamanda first in masters and all we have done lot of experiments, and now we cannot remember all the experiments we did, and there we do gram positive and gram negative bacteria. Next topic vanda, it's about the cell wall. Okay, cell wall. This cell wall students, what you have to understand, cell wall can be non-cellulosic. Okay, it is need not be made of cellulose, right? Does it have to be made of cellulose? No, in prokaryotes we have studied that it is non-cellulosic. And what do? Mainly they will determine the shape of the cell. Correct? Yes ma'am, they will determine the shape of the cell because they are the one who decides about the rigidity of the cell. Correct? Yes, no, it's just like nama skeletal system madri. They provide a framework. They provide a framework. Now students, the cell wall can be made up of your peptidoglycan, or we say in other words murin. So here you can remember they can be made up of murin, or we can say in other words peptidoglycan. In RK bacteria we have seen that this instead of murin they have pseudomurin, right? So this is in U bacteria. We have seen that in your RK bacteria, RK, okay, we will have pseudomurin. Okay, we will have pseudomurin. Clear? So like this different types of constitutions will be there who is working for it. Okay, in the peptidoglycan one, the students have already taught you that this peptidoglycan is made up of actually your NAM and your NAG. Okay, hey ma'am, what? Okay, N-acetyl glucosamine, N-acetyl muramic acid, that is called NAG and NAM. So it is made up of repeating units of NAG and NAM. That is the structure. Okay, that much you don't have to break your head, but just remember this much. Okay, remember this much is important. Okay, last one the plasma membrane. So what we will do, we will shift. Oh, this color will go now. Okay, we'll color it again. Okay, I don't like this gray color. And this will also go. What is this? Okay. Okay, I'm keeping it here. Okay, and I'll shift the glycocalyx. I'll shift it here. Okay. Yes. Okay, now we will write it here. Okay, done. Now here your plasma membrane. Plasma membrane. Plasma membrane, one the students, what do we know? Ma'am, they are selectively permeable. Okay, and they are the one students who is interacting with the outside world. Others are not ready to do anything. Okay, they are the ones, they are interacting. With the outside. Okay, interacting with the outside environment, right? Interacting with the outside environment, and from the plasma membrane only we have lot of extensions, right? Plasma membrane only students, we have lot of infoldings that are there, like your mesosomes. Okay, mesosomes irukku, surface structures are there, நம்ம flagella irukku, இது எல்லாமே is because of the plasma membrane. Clear? This is the basic thing that you have to understand. Here I will just add on with the gram positive and gram negative. Just remember, நம்ம bacteria, okay, two categories, we will divide them into gram positive and we will divide them into gram negative. Okay, gram positive and gram negative. Gram positive na, they will take up the stain. Okay, they will take up the stain. Okay, their layer structure will be little different, that is the reason why they are taking up the stain. And then gram negative means they will not retain the stain. Okay, they will not retain the stain, and we usually do this, that is nama pigmentation vande illana staining vande nama usually crystal violet and the madri pigments use panni we do this. Okay, so this is your entire this one. I hope it is not confusing. Is it too confusing? I hope it is, you know, decent enough for you to understand. Okay, so that is done. Now we will slowly move to eukaryotic cell. Okay, eukaryotic cell. I'll just get some water for myself, students, and then I'll be back. Not water, we will bring coffee. Okay, coffee mukyam bigile, right? Okay guys, eukaryotic cell start பண்ணலாமா? ஆனா eukaryotic cell முன்னாடி we have to study one more topic, one kutti topic, that is your ribosome. Why ma'am? Because ribosome is a universal organelle. It is going to be there in prokaryote, eukaryote. ஆனா the number, like we write no 70s, 80s, அது வந்து கொஞ்சம் different ஆயிருக்கும். So look here, in prokaryotes the ribosomes are associated with the plasma membrane of the cell. When I drew the diagram, if you all noticed properly, you would have noticed that I drew it sticking to the cell membrane, right? Here, look here. Look here, very close to the cell membrane, or almost like sticking to the membrane. So that's why, see, look here, with the associated with the plasma membrane, they are about 15 nanometer, okay, by 20 nanometer in size, and are made up of two subunits. All of you remember translation? Yes or no? So in translation we have studied about this, and they are made up of two subunits. One is 50s and 30s subunit, which is when present together forms 70s prokaryotes. You will be like, ma'am, ribosome is more weak than me in maths or what? Okay, maths la vande na vande romba kevalam, ana ribosome vande adikkina kevalam, ma'am. How? Okay, So the answer to that is no, because the S does not stand for addition. Okay, 50 plus 30 is 80. Ma'am, how is it 70? I'll teach you that. Okay, now look here, ribosomes are the site for protein synthesis. This we have already studied. That is where we have studied about translation in our molecular basis of inheritance. Okay, molecular basis of inheritance. Several ribosomes may attach to a single mRNA and form a chain called as polyribosomes or polysomes. The ribosomes of a polysome translate the mRNA into protein. So let's learn it detailedly. So we have our ribosome. Okay, ribosome, correct? Yes, ma'am. In the ribosome, students, one thing that you have to understand is that this is a universal organelle. Universal organelle. Okay, universal organelle. In the universal organelle, one very important thing is nama prokaryotes or eukaryote cell. Nama prokaryotes. Prokaryotes and nama eukaryotic cell. Now the ribosome we use is different, right? Here we say 70S ribosome is present. Ana in the 70S we will have one big larger subunit called as 50S and one smaller subunit called as 30S. Ana ma'am, 50 plus 30, 80 should come. So students, this S actually stands for sedimentation factor. Now what is sedimentation factor? Imagine panikonga, this is our one, centrifugation types, we are doing a sedimentation. In the sedimentation, what you will notice that at certain point, okay, we say sedimentation coefficient, we say. So in the madri, there will be some lines. Just imagine like this, okay. So the one that will, so let's say we start from 20. No, we'll start from 30. Okay, 30S, 40S, 50S, 60S, 70S, 80S. Okay. So this is our sedimentation factors. Now you will notice that one portion of the larger subunit might go and attach here in the 50S. Okay, 50S, and one more smaller subunit will go and attach like this. So this is your sedimentation coefficient, Svedberg's unit. So it does not stand for 50 plus 30 should be 80. Okay, you cannot fight for it. You should remember that the larger subunit, okay, the larger subunit and the smaller subunit are going to be based on the sedimentation. Okay, so this is going to be 30 and this is going to be 50S, and the S stands for Svedberg unit or else sedimentation factor. Coefficient also we can say. Understood? So next time someone tell you this maths is wrong, you should tell them that you're wrong. Okay, this is understood. Now we have all studied one very basic thing, students. What is that basic thing? That here we have studied about 80S. Okay, here we have studied about 60S and 40S. Okay, this we have studied in eukaryotes. Now, more than this, one very common thing that what we have studied is about translation. Yes or no? So, we have all learned that we will have one mRNA molecule. Okay, we will have one mRNA molecule, and that mRNA molecule will have one subunit like this. Okay, like this subunit will be there. Lot of ribosomes will be attached, and what they are going to do, students, they are reading the mRNA. Now, already theriyum, because reading and translation we have already studied. So, if you are a class 11 student who has not studied molecular basis of inheritance, it might be little difficult for you to understand. But if you are a NEET student, you will know this. So, nama ribosome, multiple ribosome or mRNA mele, they will be reading and the translation process nadakum. So, this entire thing, can I call it polyribosomes? Yes, ma'am, you can call them like that because many ribosomes are present. Polyribosome, illena polysome. Polyribosome, illena polysome. Polysomes. So, ultimately, in the mRNA, it is going to convert and it is going to give us what? it is going to give us a beautiful polypeptide chain. Okay, like this we are going to get a polypeptide chain. Got it? This is the main thing about ribosome, sedimentation factor or Svedberg's unit. Okay, larger and smaller ribosomes will be present. If we see, no, in endoplasmic reticulum, we say if ribosomes are present, rough endoplasmic reticulum, smooth endoplasmic reticulum, and mainly about ribosome, this is how much you have to study. Okay, this is how much you have to study. And yes, students, the function is what? Mainly they are going to do the function of protein synthesis. That is why we use them in translation. Protein synthesis. Okay, protein synthesis, that is why mainly we are using them in translation. This is super duper clear. Now we are going to start eukaryotic cell. Okay, now eukaryotic cell, students, mainly eukaryotic cell, when I tell you itself, I want you to remember this word called as compartmentalization. 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 Ma'am, mental agadu ma'am. Ivlo spelling. Correct. Compartmentalization. Correct, no? Yeah. Compartmentalization. Why? Because students, in the eukaryotic cell, avlo organelles irukku. So, all organelles will have their own border. They'll have their own field and also adanale, because there are many organelles, compartmentalization is a lot in your eukaryotic cell. Clear? Okay, now look here, in eukaryotic cell there is an extensive compartmentalization of the cytoplasm through the presence of membrane bound organelles, a cell membrane and maybe a cell wall too. Eukaryotic cell possesses an organized nucleus with an organized nuclear envelope, right? Because without a nuclear envelope, I will not call this eukaryotic cell. I am calling it eukaryotic cell because they have the nuclear envelope. Correct? Yes, ma'am. Correct. Now, look here. The eukaryotes include all protists, plants, animals and your fungi. Right? So, from kingdom Protista, Animalia, Plantae, fungi, all of them. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through the presence of membrane bound organelles, because each organelle will have their own field place. So, everyone will be like, I want my place, I want. So, adanale adhigama compartmentalization nadakkum. Okay va? Understood? Okay. In the eukaryotic cell possesses an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton. Cyto na student cell. Skeleton na what does it provide? Framework. So, cytoskeleton na it provides the framework. So, cell frame. Adada nama cytoskeleton structures. Okay. Clear? Now we are going to get detail into it, okay. Now gram-positive bacteria take up the crystal violet stain and appear purple colored. Thick peptidoglycan layer in the bacterial cell will retain the stain and lipid content is less. Okay, so basically how they are going to, basically have the stain. Gram-negative bacteria cannot retain the violet stain, 10 to 20% peptidoglycan, 20 to 30% lipid content. Alcohol use degrades the outer membrane of the gram-negative cell and makes the cell wall more porous and incapable of retaining the crystal violet. So this I already told you, you don't have to break your head. Now we'll come into the main part. Now, the detailed structure of the membrane was studied only after the advent of electron microscope. So, we are studying about what? We are studying about the cell membrane. Okay, cell membrane. It was only found, students, in the 1950s. Okay. Meanwhile, nama chemical studies on cell membrane, especially in human red blood cells. Why, ma'am? Edukku ma'am only RBC they used. Why didn't they not use any other cell? Because, students, RBC is the least contaminated cell. Least contaminated cell. Least contaminated cell. எதுக்கு? Why? Because இந்த செல்ல வந்து நியூக்ளியஸ் கிடையாது, ஆர்கனல்ஸ் கிடையாது. ஸோ அதனால அந்த செல் மெம்பரேன் பார்க்கறதுக்கு, அதை காம்போசிஷன் படிக்கிறதுக்கு ரொம்ப ஈஸியாக இருக்குது. ஸோ அதனால தான் they used RBC cells. Question can come, so make sure to remember. Enable the scientists to deduce the possible structure of the plasma membrane. So it was this RBC was the one who was helping them to find the cell membrane. What is there in the cell membrane? Because அந்த டைமில் நமக்கு வந்து RBCயில் லீஸ்ட் கண்டாமினேஷன் இருக்குது. ஸோ அதனால அந்த செல் மெம்பரேன் ஸ்டடி வந்து ரொம்ப ஈஸியாக நடக்கும். ஸோ that is why they have chosen RBC. Okay. These studies showed that the cell membrane is composed of a lipid bilayer, protein, sugar and cholesterol. Cell membrane is composed of lipids that are arranged in bilayer. So we are going to study detail of this, okay, one by one. So first let's make some slides. Okay, that is the task number 1. Okay. Now we'll study from the starting. Okay, we're going to study from the starting. That is about your eukaryotic cell. See, even if I keep notes, student, even if I keep printed notes, I don't feel satisfaction until I write and make you understand. Okay. So that is how I am. So eukaryotic cell la mainly enna nadakum? Ma'am, compartmentalization romba adhigama irukum. Compartmentalization ரொம்ப அதிகமா இருக்கும். Why? Because there are many organelles. So அதனால compartmentalization அதிகமா இருக்கும். Second thing வந்து students, they have cytoskeletal elements. Cytoskeletal elements. Okay, இந்த cytoskeletal elements na enna? Cyto na cell, skeletal na enna? Framework, ma'am. You already told that. Okay, and they also have complex locomotory structures. Correct dane? Yes ma'am, correct, very correct. Okay, இப்போ cytoskeleton na enna? Cell வந்துருச்சு. Okay, cell, அதுக்கப்புறம் skeleton na frame. Okay, frame. Okay, so cell, frame and complex locomotory structures. Then students, this entire structure also has a network of protein filaments. In the entire structure there is a network of protein filament. It's all like, you know, so many networks are there, otherwise this complex structure will not be formed. So the first protein filament is going to be number microtubules. In the microtubules word, where did we study before? When we taught about flagella. So in the microtubules will help in locomotion as well, right? It is going to help in locomotion. We can call it as MT, and they are going to help with locomotion because of the tubulin protein. Okay, locomotion. Tubulin protein na microtubules da. So tubulin protein it is going to help. Next one the nama intermediate filaments. Intermediate filaments. Either we can call it as IF. Okay. Adukapram students namakku vanda microfilaments. Illena, you have heard this word very clearly, actin filaments. Actin myosin contracting protein, so and the actin filament. So these are the different types of protein filaments that are there, okay, and they are going to help with the entire process, okay, maybe with locomotion, movement, எல்லாத்துக்குமே இந்த protein filaments வந்து maximum it is going to help. Okay, இதில microtubules will have more diameter, in the microfilaments will have less diameter. So this is the whole thing. Okay, adukapram students, we are going to come to the next part. They are saying that this membrane, this layer is made up of protein plus lipid. Yes or no? They are telling this cell membrane or the cell membrane that we were studying, it is made up of protein and lipid. And whom did we use for it? We used an RBC to understand this. We used whom? We used a RBC cell to understand it, because in the RBC cell least contamination irukum. least contamination. அதனால நம்ம வந்து RBC, and I will also write it in bracket because it is also enucleated cell. Okay, enucleated cell. So they are saying this layer is made up of protein and lipid. So let's understand each word, right ma'am? Yes, let's understand each word. So protein, now students, there are two types of proteins in this entire plasma membrane. Enna the first protein vanduchu, okay, first வந்து நம்ம intrinsic protein. என்னது? Intrinsic protein. Okay, next வந்து extrinsic protein. Intrinsic protein, அதுக்கப்புறம் extrinsic protein. Intrinsic, in, okay, so that means it is going to be, we can say it is embodied inside. Okay, embodied inside. Extrinsic na external, peripheral proteins. இது வந்து remove பண்றதுக்கு ரொம்ப easy, இது வந்து remove பண்றதுக்கு ரொம்ப கஷ்டம். Because inside la irukku. So inside in, let's say if I want to pull out your larynx, is it possible? No, I can't. But I can pull the skin from here, right? So அது மாதிரி inside la இருக்கிறது pull பண்றதுக்கு ரொம்ப கஷ்டம். ஆனா இது வந்து pull பண்றதுக்கு ரொம்ப easy. Okay வா? Yes? Oh, this mic. Okay, so intrinsic protein na partially illena totally buried ஆயிருக்கும். Partially illena totally buried ஆயிருக்கும். Extrinsic na they will be external. Okay, they will be hardly buried nu சொல்லலாம். Okay, now next layer வந்து lipid. Okay, we'll go detail to this, but first you understand this much. Okay, again I'll teach you more about it, but understand about this much. அதுக்கப்புறம் students நமக்கு வந்து lipid. Lipid வந்து bilipid layer. Bilipid layer னு சொல்லுறாங்க. இல்லனா phospholipid layer. Phospholipid layer. This is what they are saying. So this is the general of it. Now we have to understand detail of it. Anna, let's just look at this lipid structure. So it will have a head. Okay, it will have a head and it will have tail. Okay, so idu vandu head, idu vandu tail. Enna tail, enna head, what is the use? Adellame next slide la we will study. This much is clear, students? Okay, so once again I'm telling eukaryotic cell வந்து maximum compartmentalization இருக்கும் because அவருக்கு வந்து organelles அதிகமா இருக்கு. So அதனால எல்லாமே border இருக்கும். அதுக்கப்புறம் they have cytoskeleton elements. In the cytoskeleton elements builds the framework of the cell and also complex locomotory structures. They also have network of protein filaments like your microtubules helping in locomotion. Why? Because that flagella formation tubulin protein function will be done by the microtubules. அதுக்கப்புறம் intermediate filaments and microfilaments or your actin filaments, which are again going to help in the movement. Okay, then students they are telling that in the plasma membrane la mainly protein irukku, lipid irukku. These are two main things. Protein வந்து intrinsic protein and extrinsic protein. Intrinsic protein na partially or completely buried ஆயிருக்கும். Extrinsic protein na externally இருக்கும், easy to remove. Lipid layer na bilipid layer இல்லனா phospholipid layer னு சொல்லுவாங்க. அந்த lipid structure பாக்கும்போது ஒரு head இருக்கு, ஒரு tail இருக்கு. Dandana done. Yes or no? Understood this much? Who will not understand this much? You tell me. Huh? I've not even taught the full. I've just started with the very, very basic. Yes or no? Very, very basic we have started studying. Okay, understood this? We have to study more, but then tell me if you understood this or not. Okay, let's move forward. Okay, next one. Students, நம்ம இப்போ வந்து structure draw பண்ணலாம். Okay, ma'am, structure draw பண்ணலாம். Okay, this is the structure. Yes, but here I have to draw borders. Okay, clear border? Almost clear. Now again one problem is there. We only have problems, students. We have less solutions. This is okay. Yes, half of the problems are sorted. Okay. Yes. So, this is our lipid layer. Okay, bilipid layer. We are just drawing it. Okay. Now, look here. Okay, I want to draw a better border because I don't think this is visible for you. In the border one, the important students, because in the border la thaan nama proteins draw pannam. So, adinale this border is important. You'll be like, ma'am, why are you drawing border, ma'am? Okay, 1 minute. So, slow process, but the learning is going to be beautiful. You will enjoy it. Okay. Yes, this is the layer. Okay, done. Now it's better, right? Okay. Now, first we will draw, students, some proteins. Okay. Namakku vanda, there are going to be some proteins which are attached like this. Okay. Which are your extrinsic protein. Correct, ma'am. Extrinsic. It is sitting outside. Okay. It is in the outside. Okay. Then we will have some proteins which are in the madri. Okay. It will be in the between. Okay. Between irukum. That is your intrinsic protein. And then we will have one big tunnel-like protein going from one side to another side completely. You'll be like, hey? Yes, that hey is correct. Okay, they'll be going completely from one side to another side. Okay, these are called as transmembrane proteins. Now, try and understand, nama lipid layer, I told you they have a head and they have a tail. Correct. So, I haven't drawn the tail. So, now I am drawing the tail. Okay. This is the tail. Okay, wait 1 second, students. I'll draw first the tail, then I'll draw this transmembrane protein. Okay. Okay, this is the tail. Okay, like this. So, this is the head and this is the tail. Okay, done? Yes or no? Okay, this is done. Now I'll draw the transmembrane protein. Okay, this is your transmembrane protein. கடவுளே என்னாச்சு? Okay, yes, this is the transmembrane protein. So now just look here. This is your bilipid layer. In the bilipid layer, students, namakku vandu oru head irukku. Yes, ma'am, correct. Namakku irukku. Okay, there is one head. Correct, this is the head. Okay, and then we have these two tails as well. This head, students, this is a polar head. Polar head na enna? Okay, this is a polar head. Okay, it has charge and this head will get attracted to water. It loves water. So adanala in the polar head vanda it is hydrophilic. Polar head vanda it is hydrophilic. That means it loves water. Why so? Because water charge paakumbodhu you have H and you have oxygen. In the oxygen vande electronegative. Okay. In the hydrogen vande electropositive. So this will get attracted like this and the entire thing, like electronegativity will be there. So adanale this is also charged polar body, right? So this will become polar head where it will love water. Adukapram we have non-polar tail. We have non-polar tail. That means this fellow hates water. It has fear to water. So idinale this will be hydrophobic tail. Okay, hydrophobic tail. Clear? So one loves water, one hates water. So adinale what is going to happen here? What is going to happen here? The water from here has to be accepted, right? Polar head. Polar head. Clear? Yes or no? This hydrophilic, hydrophobic, okay va students? Now understand, nama protein mele, if there are like this receptor kind of structures, these are your glycoproteins. What are they? These are your glycoproteins. Why? Because they are on the proteins, okay, and they'll be helping for that. Then if they are present on the lipids, nama vanda the glycolipid nu solluvanga. Okay, glycolipid we will call it. Now, coming to a very important conversation, okay. But let's say students, I'm telling this is a bilipid layer, this is a phospholipid layer. This lipid means what? Is very like movable, okay. It's very movable and all of that. Now imagine in the lipid, let's say this lipid wants to go here. Is it possible? Yes ma'am, it is possible, okay. It can move from here to here and can move from here to here. Both of them can move that side and this side. Okay, correct. Let's say this wants to go here. Let's say this wants to move here. And this wants to move here. That is also possible. Yes ma'am, because they are little fluidy, they will move off. There is no difficulty in it. But imagine from here to here, one person wants to move. Let's imagine this fellow wants to move here. Is it possible? It is very, very rare. Okay, this is very, very rare. This is the rarest occurrence. Okay, and we call this movement as flip-flop. We call this what? Flip-flop. So, lateral movement nadakum because in the, idu vandu fluidy aayirukku. So, adanal inge move pandrathu mudiyum. Ana, from one layer to another layer vandu konjam difficult. Ana, it is possible. It is rarest, but it is possible. That kind of movement, we will call it as flip-flop movement. Flip-flop movement. Okay. Now, coming to a very important part. Imagine, I told you this plasma membrane is very, very selective. Correct. This membrane is very selective. Since it is very selective, selectively permeable it is, so many things will want to move that side and this side. Okay. Now, I cannot allow movement of everything. Now, who will move, how they will move, it's important. Now, you will be thinking, ma'am, can you label all of this? Of course, yes. Okay. Let's label all of it for you to understand better. So, idu vandu nama glycoprotein. What is it? Glycoprotein. Where have I placed it? On the protein. Okay. Idu vandu nama extrinsic protein. Correct? Yes ma'am, correct. Idu vandu nama intrinsic protein. It's partially or almost fully embedded. இது வந்து ரிமூவ் பண்றதுக்கு ரொம்ப கஷ்டம் ஸ்டூடெண்ட்ஸ். இது வந்து ரிமூவ் பண்றதுக்கு ரொம்ப ஈஸி. ஓகே, இது வந்து நம்ம glycolipid. இது வந்து நம்ம glycolipid. Okay. Now, this big fellow who is acting or who is going to behave like a tunnel is called as transmembrane protein. Transmembrane protein. And this transmembrane protein is usually called a tunnel because it will allow the movement of certain things. What the plasma membrane will be like hesitant to allow the movement, nama vanda tunnel but use panni we can move it. Now like imagine in certain countries certain things are not allowed but you want to transport it there. What will you do? You'll go by ship. You will go by water means, okay, where the checking is not there properly, you will use a tunnel system to transport something. Anda madrita, I will use this tunnel system to transport anything which is necessary for me. So, like that, we can do transport system. Clear? Now, coming to a very important part, students, in this layer, vanda romba fluidy irukku. Correct, ma'am, romba fluidy irukku. So, there are spaces in between. So, that spaces, you will notice that some cholesterol particles are present. What will be present? some cholesterol particles will be present, and this cholesterol particles actually provide the stability. Cholesterol particle வந்து என்ன provide பண்றாங்க? Stability. இந்த diagram students இப்போ பார்க்கும்போது அவ்வளோ clear-ஆ கிடையாது, but ஆனா in your PDF, in the description box when PDF comes, it will be very clear for all of you. So இதுதான் நம்ம cholesterol. What is cholesterol doing, students? I'll tell you a very simple example. Nama chair, okay, in our chair and all, like in the wooden chair, sometimes when we sit, it will go one side down. So what we will do, we'll fold a piece of paper and insert it there. Correct தானே? அந்த insert பண்ணும்போது அந்த chair வந்து stabilize ஆகும். அந்த மாதிரி தான் இந்த cholesterol. அந்த spaceல இந்த cholesterol present ஆயிருக்கும். So it is going to do the job of stabilization. What is it going to do? It is going to act as a membrane stabilizer. It's going to act as what? A membrane stabilizer. Okay, va? This much is clear. So very simple, I have taught you. Now again we'll read NCERT, don't worry. So when you read NCERT, you will have more better idea about it. So this much is understood. Cholesterol will act as a membrane stabilizer. Now coming to a very, very important topic, how the transport is going to happen. Ipo, let's say I want to transport something. How will the transport happen? Let's draw it elsewhere. Okay, let's duplicate this and let's erase. Oh. Okay, we have duplicated it. So let's erase this. Okay. Now imagine, students, idhe madri I'm just drawing two lines. Okay, just two lines I'm drawing, so the work becomes easier. This is the first bilipid layer. Okay. We have to learn about fluid mosaic model and all. I'll teach you. I might be teaching you in a different order compared to other teachers, but don't worry, everything will be covered. Okay. So this is the two bilipid layers. Now you tell me, transport has to happen, and let me just draw the beautiful tunnel here. Okay, we have the beautiful tunnel here. Okay, now you tell me, if something has to be transported, let's imagine firstly vanda namakku vanda oru non polar substance transport பண்றதுக்கு எல்லாமே students, everything will move from a region of higher concentration to lower concentration. Yes or no? Yes ma'am, very true. So if a non-polar substance has to be transported, it will be transported through the membrane. Yes or no? Why? Because we have more non-polar tails. We have non-polar tails, so the transport will be easy. Right? Transport will be easy. So the non-polar one can transport from a region of higher concentration to a region of lower concentration with the help of a very simple process called as simple diffusion. What is it? Simple diffusion. Okay. Correct, ma'am. From higher region to lower region, using simple diffusion, I have transported it. Anna, imagine I want to transport a non-polar substance. I want to transport what? A non-polar substance. If I want to transport polar substance. If I want to transport a polar substance, the problem is what, students? Tail or head mattunda polar. Head mattunda polar. So, adanaale oru polar substance vandu inge stop agum. Correct dane? Only head is polar. Rest everything is non-polar. So what I will do? I will use a tunnel to transport it. So I will use this non-polar substance to be transported with the help of tunnel from a region of higher concentration to a region of lower concentration. Now this will be facilitated diffusion. Facilitated diffusion. Understood? So, polar substance vande, if I want to transport, adu vande inge vande stuck agum because only the head is polar. So, adanala I have to use another medium to do it. So, that's why I'm using this tunnel, I'm using this transmembrane to transport the polar substance. Done, ma'am. Now, imagine I want to do something against the nature. Against the nature means what? I want to transport something from a region of lower concentration. Okay? From a region of lower concentration to a region of higher concentration. You will be like, ma'am, how, ma'am? This is so impossible. Okay, because you're doing against the nature. Right? So, adanala konja impossible a irukku. But I want to do it. So, if I want to do such a kind of thing, students, then the only medium for me to do this is with the help of energy. With the help of what? ATP, or with the help of pump. All of you remember sodium potassium pump that I taught you in neural control and coordination? That time what was happening? The pump was helping us to push it out. No Kia. 3 sodium outside, 2 potassium inside. For that movement, I needed energy. So if I want to do something like this in the opposite direction, that is from a lower concentration to a higher concentration, then for that, I will need a energy utilization. That is, I will need an active transport to work where the energy is utilized to transport it from lower concentration to higher concentration, or with the energy I will need a pump as well, a voltage gated pump who is going to do the job for me, just like a sodium and potassium pump. Is this clear? Now students, if water molecules are going to move, okay, remember, if water molecules are going to move again from a region of higher concentration to a region of lower concentration, then it will move with the help of process called as osmosis. Okay, with the process called as osmosis. So, idellame passive transport da, because here and all I am not using energy. So, all this is technically passive. Right, it is all passive transport. That is, without energy the transport is happening. Now for that I will need energy, which becomes active. I hope this much is clear for all of you. Okay. Now students, this lipid layer we call it as fluid mosaic model. It was given by actually Singer and Nicholson. Okay, we call it what? Fluid mosaic model. This is actually a very, very detailed topic, okay? Like in our CSIR net book, this itself is like one chapter by itself, okay? Fluid mosaic model. Fluid na students, I will just break down the word for you. Fluid na nama lipid layer, okay? Fluid na nama lipid layer. Mosaic na nama protein. Students, it's like how in this Paisam we keep, no, when we keep paisam, we put cashew nut, dates, almonds, and the madrita. Proteins are like that, okay? Fluid mosaic model. This fluid mosaic model was given by whom? It was, ideated by Singer and Nicholson. Singer and Nicholson. These two people gave the fluid mosaic model. Clear? Understood? Now functions and so much we have to study, that we are going to study slowly. Okay. Yes, cholesterols are going to be present, which will act as a stability, okay, to provide stability for the membrane. Clear? Okay. How much lipid is there? How much lipid is there? Somewhere around 40%. Okay. So here, where do I write? Okay, I'll write it here. Yeah. So, lipid students, lipid plus protein combination this is, no? Protein is around 52% and lipid will be the rest. How much is it? 48%. Okay, 48%, rest will be lipid. Okay, and also remember this in your proteins as well. Excuse me. Sorry, someone is thinking of me. Intrinsic protein, difficult to extract. Difficult to extract or remove, we can say difficult. Okay. Extrinsic protein, it's much easier. Okay, easy. Got it? Yes, ma'am, got it. Okay. Now we have to study the function of this. So we will study one by one. Okay, we will study slowly. Don't worry. So this is your eukaryotic cell. This is the membrane. This is the transport. Now let's come back here. The lipid component of the membrane mainly consists of phosphoglycerides. Okay, what they will have? Phosphoglycerides. These have a polar head group and 2 hydrophobic hydrocarbon tails. What will be there? tail will be there, head will be there. Okay, one tail will be there, two tails will be there. Okay, these are the saturated fatty acids, unsaturated fatty acids. All this you don't have to break ahead. Phosphate will be there, glycerol will be there. That is what the head is made up of, and it is hydrophilic head. Okay, it is a hydrophilic head. Now, lipids are arranged within the membrane with a polar head outside, facing outside, and the hydrophilic tail facing inwards. This ensures that the non-polar tail of the saturated hydrocarbon is protected from aqueous environment, and only the head is in contact with the water. So here you can see aqueous internal. So this is how it is. Tail is put inside so that it doesn't have to face the aqueous that is water condition, because it doesn't like water. Okay, it doesn't like water. Now, along with the lipids, they have proteins and carbohydrates which are embedded in the lipid layers. So in human beings, the membrane of an erythrocyte, okay, erythrocyte has approximately 52% protein and almost 40% lipids. Okay, I have written here 48%. I wrote the whole balance, so here we will write 40%. Okay, 40% lipid. Okay, now here, depending on the ease of extraction, membrane proteins can be classified into integral protein or peripheral protein. Integral na intrinsic, peripheral na extrinsic. Okay, wow. Now, peripheral proteins lie on the surface membrane, while the integrals are partially or totally embedded into the membrane. Fluid mosaic model is the most recent model proposed by Singer and Nicholson in 1972. According to this model, the membrane is a quasi-fluid in nature, that means like a semi-fluidy nature. This ability to move within the membrane is measured as its fluidity. Okay, to move within the membrane, it is measured as its fluidity. Now, the fluid nature of membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. So this is the basic function that we have to study. What is the function of this membrane? What does it do, students? First one it helps in nama cell growth kaga help pandranga. Adukapram formation of intercellular junctions. All of you remember cell junctions? Okay, cell junction. It is helping for cell junction. Adukapram it is helping for secretion, endocytosis, and cell division. It is helping for all of this. Okay, that is the function. So endocytosis, here you can see, the particles are entering. It is engulfing, and then the vesicles are there. So outside the cell, endocytosis. This is the cytoplasm. Endo means coming inside. Okay, cytosis is happening. So endocytosis is happening. Now you can see cell division also, it is going to help. Okay, if it doesn't help, it cannot move forward. Now, the arrangement of lipid and protein is like a protein iceberg in a sea of lipid. Namakku vandu paakumbodhu, nama protein paakumbodhu, we will think that okay, only this much protein is outside. Right, we will think like that. But Anna, if you go inside, you will notice that it is a bigger structure. Just like how we talk about the iceberg. Okay, we say no, oh, it is just tip of the iceberg. Like when I'm saying a gossip or when I'm telling something, I'll tell, oh, that is only just a tip of the iceberg. More detailed, deep conversations have to happen. Andamarida. So we will notice that these proteins are the icebergs in the sea of lipids. Lot of lipids are there. In that full of lipids, nama proteins on the just a iceberg madri. Okay, clear? So here you can see cholesterols are there, lipids layers are there, proteins are there. Now, neutral solutes, now how they are talking about the movement, whatever I taught you, how they are moving. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower concentration. First we have spoke about neutral solutes. So neutral solute, students, automatically they will move from region of higher concentration to lower concentration with the help of simple diffusion. Okay, simple diffusion. Adukapram, water may also move across this membrane again by the process of osmosis, which I already taught you. Now, as the polar molecule, so non-polar, your neutral solute pass. Okay, osmosis, water molecules also pass. Adukapram, as the polar molecules cannot pass through the bilipid layer, lipid bilayer, so they have to they require a carrier protein. They require what? A carrier protein or a transmembrane protein to do the transport. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. So, in the topic avlo detail they have not explained. Anna, I have already explained you that they will use a transmembrane protein, or the other word you can use carrier proteins, for the movement. Okay, for the movement they will use these two. Clear? Now look here. Let's say we have to move it from a fewer ions to a higher concentration, then what will happen? A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. Such a transport is an energy dependent process where we are going to use the ATP. So ATP is utilized and it is called the active transport, that is like a sodium potassium pump. Is this clear? Okay, whatever I explained, I am just making you read NCERT, so it is super duper clear in your head. Many molecules can move briefly across the membrane without any requirement of energy, and it is called as passive transport. Without using any energy, some of the particles can move up and down, that is called as your that is called as your passive transport. Now, cytoplasm, one second students. Before we go to cytoplasm, we will finish off the cell wall, okay? Then we will go to the cytoplasm. So, cell wall, okay? This is super duper clear. Any doubts you have? Should I write the functions for all of you, or is it okay? If you want me to write the functions, I can write the functions, okay? Students, listen, you will also hear the word exocytosis, where the cell vomiting, removal of it. Okay, we will also listen about cell drinking, cell eating. That is what we do in the cytosis, endocytosis. Okay, phagocytosis means cell eating. Okay, phagocytosis is the exocytosis, is the removal of the cell. So these words also just keep in your mind. I am telling you now, so that you know you don't, first time when you see somewhere, you will be like, oh my god, Gopika Mam didn't teach us. Okay, so you don't be confused about all these words. Okay. I'll write it here only. Functions mainly, see, we already understood it is doing the interaction and all of that, but other than that, mainly I will tell you, they can do endocytosis, engulfing, okay. They can do phagocytosis in this. Phagocytosis. Are you all marking your attendance? Quickly smash the like button, okay? Quickly smash the like button. Pinocytosis. Okay, this is cell eating. This is cell drinking. Okay, exo means outside, throwing it outside, so removal. Okay, removal. Clear? Removal. This is there. Understood? Active transport, passive transport also you people would have understood already there. I have explained it in this one slide. So I hope this one slide will help you. With that, lot of explanation is also there. Now we will study cell wall. Okay, cell wall. Yes, cell wall. Okay, cell wall means what, students? It is the outermost covering of the cell. Cell wall protects the cell from mechanical damage and infection. Cell wall has a cell to cell interaction. Okay, cell to cell interaction. Now I am going to teach you a very important phenomena. After that, again we will read NCERT. So look here. Okay, this is little grey only. Let it be grey only. So we will have cell wall I am drawing, okay. This is one. Inside this, oh no, this is very pathetic. So in Sunday, I am going to keep a human physiology one-shot revision. So make sure that you are attending. Okay. This is done. Okay. This doesn't look nice. I don't know why. Okay. So just look here. This is the cell wall formation, student. This is the primary wall. This is what? The primary cell wall. Okay. And this is the actual cell wall. So this primary cell wall will allow the cell wall to grow. Okay, it is a more flexible layer, so it will allow the cell wall to grow. And then again, after that, what will happen, students? Just look here. You will have again primary cell wall. Okay, actual cell wall. And in between this, there is going to be one more layer. Like this, one more layer. Okay, now you will notice that the first layer would have slowly started, diminishing. Okay, diminishing. Now, what did you understand? The first primary layer, students, this primary wall allows the growth of the, allows growth of the cell wall. Okay, this is the first stage. After that, what will happen? This will be the primary wall. This primary wall will be diminishing. You can see that I have drawn a few black lines. Then this green color one is going to be the secondary wall. In the secondary wall one, the students, it will not allow, and the primary wall, it will not allow like the primary wall to grow. So the cell wall will be done with its growth. Okay, cell wall cannot grow anymore because in the secondary wall one, it does not allow any space for any growth. Okay, now here, this to this is happening. Okay, this to this, it is happening. So this is your cell wall formation, just a bit of it. Now remember that when two cells are coming together, okay, when two cells are coming together, you will notice that two cells will be connected, or they will, let's say, like how I taught you in cell junction, no, they will communicate with each other with the help of a portion which is called as your plasmodesmata. What is it called? Plasmodesmata. Okay, plasmodesmata. Understood this? Okay, they are actually cytoplasmic strands. Plasmodesmata. Cytoplasmic strand. Okay, so they will do all the exchange with the help of what is that structure called as plasmodesmata. Now students, when two cells come very, very close to each other, they will form a middle lamellae. Okay. So I drew two different color cells. I'm capable of doing that. Okay. Students, how beautiful is our zoology chapters. Okay, when I teach botany chapters, I realize no, as a teacher, I shouldn't have partiality to one subject, but I do have partiality to zoology. Okay, like this. When middle lamella will be formed when two cells are together. Okay, middle lamellae. And students, they can be formed with calcium and magnesium. Okay, they can be formed with calcium, magnesium, pectate. Calcium, magnesium, pectate, they can be formed. Middle lamella. Again, this is the basic. Again, we will see NCERT as well for you to have a better clarity on what we just studied. So reading, when I read NCERT, you will see this is explanation part, but for you to understand more detailedly, I will tell you about your NCERT as well. Okay. Okay, let's go. Dedication you see, students. Okay, yeah. Everything is black, right? This is not black. Huh? Okay. Now look here. Cell wall is made up of, okay, this is important. You should know what the cell wall is made up of. It is made up of cellulose, hemicellulose, pectin and proteins. Okay. Cell wall will be made up of multiple things like what and all? Cellulose, hemicellulose, pectin and protein. Now, cell wall has three parts. There is the middle lamellae, okay, primary and secondary wall. So, these are the three regions I taught you separately. So, you can see primary wall will allow the growth of the cell wall, like it can stretch, it can be a little more flexible, but secondary wall will be like, no, you cannot move anymore. We are done with the growth, okay. So, middle lamellae is made up of calcium and magnesium pectates, okay. The softening of ripe fruits is due to the solubilization of this pectate. Middle lamellae helps in attachment of two adjacent cells. I told you, know, when two cells comes very close to each other, they will help in that. Middle lamellae will help in both the cells to be attached. It's just like a common wall. Okay, it's like a common wall. Just imagine, students, you and your neighbor house will have one common wall. If the site is like, let's say, both of you are in a single site, you have taken half and half, then both will be attached like that. Correct? Yes or no? Yes, ma'am, it will be attached like that. Okay, so it's a common wall between your adjacent rooms. The cell wall of a young plant, the primary cell is capable. See, the primary wall is capable of growth, which will gradually diminish as the cell matures, and the secondary wall is formed on the inner towards the membrane side of the cell, okay, towards the membrane side of the cell, and it will not allow any more growths. It will be like enough of your growth, enough. Secondary wall is produced in some mature cells when it has stopped growth. It has three layers like S1, S2, S3, which is little detail for you, but just understand S1 layer, first layer, S2 and S3 layer, they will not allow any more growth of the cell membrane, okay, they will not, sorry, cell wall. Now, plasmodesmata are cytoplasmic bridges between the adjacent cells which develop a minute pore of their cell wall. It forms a controlled passage for small size molecules between the adjacent cells. All of you remember gap junctions I taught you in animal tissues? And the madrita, chinna exchange kaga, oru nama vanda window, like how we make windows, no, between two houses to talk to each other. What did you make today? Curry. And the madrita, one small cytoplasmic bridge is there so as to transport things from here to here. Okay. So it's a very small cytoplasmic bridge that is called as plasmodesmata. Okay. Plasmodesmata. Okay. They're asking you to find a mistake here. If any of you found a mistake, let me know in the chat box. Okay. Let me know in the chat box. Okay. 1 second, students. Found any mistakes? Okay. 1 second, students, just give me a minute. Interestingly, continue panlama. So I told you cytoplasm we will do after that. So that's why we are going to start cytoplasm now. So cytoplasm na enna nama normal or cell draw பண்ணும்போது, even if I draw one circle like this, even if you are a very bad okay artist or drawer okay ஆனா கூட ஒரு செல் வந்து draw பண்றதுக்கு ரொம்ப ஈஸி. So normally நம்ம செல் draw பண்ணும்போது இவ்ளோ ஒரு circle அதுக்கு அப்புறம் nucleus and this whatever semi fluid that we are seeing where all the organelles are going to be suspended on the place the cytoplasm. Okay, so cytoplasm is a semi fluid semi-fluid matrix present in both prokaryotes and eukaryotic cell. It is made up of water, that is 80 to 85% of water. The remaining constituents are protein, that is 10 to 15%, lipids on the 2 to 4%, and polysaccharides and nucleic acid on the 1% each. Okay, so cytoplasm la idella irukku students. namakku vandu first we have already studied namakku cell wall irukku, cell membrane irukku, adukku appuram oru there should be a surface or a matrix where all the organelles can sleep around, you know, andha cytoplasm. So அந்த cytoplasm la வந்து நமக்கு semi fluid matrix da water இருக்கு 80 to 85%. Matrix means mainly it's going to be water. Okay, அதுக்கு அப்புறம் நமக்கு constituents, that is proteins are going to be 10 to 15%. in the light வந்து familiar. Okay, 10 to 15%, lipids on the 2 to 4%, நம்ம polysaccharides on the 1%, nucleic acid on the 2%. Okay, va? Okay, now here you see, all the organelles are suspended in the cytoplasm. அது வந்து தெரியும்ல? Okay, all the organelles are suspended in the cytoplasm. Any molecule entering the cell has to pass through the cytoplasm. It is the main arena of cellular activities. Main arena of cellular activities என்ன நம்ம சொல்லுவாங்க அட்டா. அட்டா ஒரு actually கன்னடா இங்க ரொம்ப ஃபேமஸா இருக்கிற வேர்டு. இப்போ let's say all of your friends, okay, from school days you know each other. So from your school days if you know each other, then it is very common that you will have a place where you always go. So maybe நம்ம ஸ்கூல் பிளே கிரவுண்ட் இல்லைன்னா ஒரு பார்க், ஸோ அந்த ரீஜன் தான் அரீனா இல்லைன்னா அட்டான்னு சொல்லுவாங்க. ஸோ the main place where all the cellular activities are going to happen is going to be the cytoplasm. Okay, main arena of cellular activities in both plant and animal cell. The cell organelles exchange materials to the cytoplasmic matrix. Like sometime back only we studied about Plasmodesmata, cytoplasmic bridge, where the cells will be like, okay, they want to exchange something smaller ions, and the exchanges and the cytoplasmic bridge, using a bridge they will transfer. Okay, so yes, we have understood that. Now we are going to a very important topic which is called as endometrium system. Okay, sorry, endomembrane system. Endomembrane system is very important, and here you are going to study certain organelles which are connected to each other. At the same time they will have similar functions. Okay, so on the 4 organelles in the endomembrane system we are going to study. Okay, one second students. We see அங்க சைக்ளோன் இருக்கு, அதனால பெங்களூர்ல வந்து குளிர் அதிகமாச்சு students, and also it is raining also, like the weather is very very dangerous weather. Okay, so please let me know in the comment section if all of you are fine, like all of your families are also fine, because I really wanted to check on all of you. So I had asked in the telegram as well, is everyone fine? Most of them told me yes ma'am, everything is fine. Okay, now why each of the membranous organelles is distinct from each other in the terms of structure and function. Okay, and function, even though they are distinct, many of these are considered together. See, many of these are considered together as an endomembrane system. Okay, endomembrane system. I want all of you to mark your attendance, and the notes will be shared in the description box. Okay, because their functions are coordinated. The endomembrane system includes endoplasmic reticulum. So this is our first organelle. அதுக்கப்புறம் நம்ம Golgi apparatus, இதுதான் நம்ம second organelle. அதுக்கப்புறம் நமக்கு இருக்கிறது Lysosome, இதுதான் நம்ம third organelle. அதுக்கப்புறம் இருக்கிறது நம்ம vacuole, fourth organelle. Okay, since the function of mitochondria, chloroplast and perioxysomes okay are not coordinated with the above components, these are not considered the part of an endomembrane system. Mitochondria no, chloroplast கிடையாது, and peroxisomes. Okay, so there are these structures which are called as students நம்ம micro bodies னு சொல்லுவாங்க. இது வந்து it is present in both. Okay, and these micro bodies will do this that is what is the breakdown. Okay, H2O2 breakdown on the to H2 and O2. Why? Because H2O2 is very toxic for the cell. Okay, they are very, very small, tiny bodies. I'll teach you in the further. Okay, they are small, tiny structures. Okay, which are going to help. Okay, which are going to help. And students, here look at it. In the mitochondria, chloroplast and peroxisomes, it does not come under the endomembrane system. Why? Because they do not perform the same function as the other people. Other people who endoplasmic reticulum, Golgi apparatus, vacuoles and your lysosomes. Somewhere or the other they are doing packaging, they are doing some job of the same. So அதனால அது எல்லாமே ஒரு சிஸ்டம், ஒரு கமிட்டி மாதிரி, ஒரு கம்யூனிட்டி மாதிரி. இப்போ எல்லா we need to தமிழ்நாடு ஸ்டூடெண்ட்ஸ் or எல்லா கோபிகா மேம் ஆர்மி all the GMAs are like, okay, we watch Gopika Mam's class, okay, we listen to whatever Gopika Mam says. And the madri system. And the madri, there are a lot of other students who do not listen. So two parties. Okay. So that is what here they are trying to say. Okay, now we will understand. Just look at the structure, students. What and all can you see? You can see a smooth endoplasmic reticulum. Yes, ma'am, I can see a smooth endoplasmic reticulum. So just imagine, students, this is our nucleus. Okay, this is our nucleus. Okay, from the nucleus, there will be we'll have our endoplasmic reticulum, which is very closely connected to it, and then there will be some places which is your rough endoplasmic reticulum, that is where the ribosomes are going to be present. Okay, ribosomes present இருக்கிற area, அந்த area தான் நம்ம rough endoplasmic reticulum னு சொல்லுவாங்க. And now the place where the ribosomes are not present, that will be called as smooth endoplasmic reticulum. Okay, so just look here. இது vacuole, இது lysosome, and here we have Golgi apparatus, and we also have rough endoplasmic reticulum, smooth endoplasmic reticulum. So understood the difference, no? Smooth endoplasmic reticulum na ribosome கிடையாது. ஆனா rough endoplasmic reticulum na ribosome will be present. So if ribosome is present, I had taught you sometime back only, ribosome it helps for protein synthesis. So அந்த மாதிரி ஃபங்க்ஷனுக்கு நம்ம rough endoplasmic reticulum will be able to do it. Okay, now let's start with the first one. Okay, look here, students. Structure of your endoplasmic reticulum. Electron microscope study of eukaryotic cell reveal the presence of a network of reticulum or tiny tubular structures scattered in the cytoplasm, that is called as endoplasmic reticulum. Okay, என்ன சொல்றாங்க? After the finding of electron microscope, நம்ம cell பார்க்கும்போது we understood that there is a network of tubular structures, and the tubular structures are called, and they are present in the cytoplasm. Obviously they are suspended in the cytoplasm, and they are called as endoplasmic reticulum. Okay, now hence ER divides the intracellular space into two distinct compartments, luminal space and extraluminal space. Now example, நம்ம draw பண்ணலாம். Okay, இதுதான் நம்ம nucleus. Okay, this is our nucleus. So it will be attached like this to the nucleus. Okay, அதுக்கப்புறம் இதுதான் நம்ம this is one. Okay, this is our endoplasmic reticulum. Correct, students? So in the endoplasmic reticulum there, whichever region I am going to shade now, okay, whichever region I am going to shade now, yeah, this region, this pink color region, can you see? And the pink color region that nama luminal space. Enna ma'am? Luminal space, okay? Intracellular space, okay? This is our luminal space, okay? In the inside place, the luminal space, okay? And whatever is outside, adu extraluminal space. Okay? So, look here. Luminal space, that is inside the ER, and extraluminal space, that is the cytoplasm. So, imagine panikonga students, idu let's draw it inside a cell. Okay, let's draw it inside a cell. Okay, this is a cell. Imagine panikonga. Okay, here I will erase this, all this. Okay, so imagine this is inside a cell. Yes, ma'am, it is inside a cell. So, இது luminal space, Okay, inside the ER, அது வந்து luminal space. And in the cytoplasm, whatever is not there, like whatever is not inside the endoplasmic reticulum, அது வந்து extra luminal space. Okay va? Yes ma'am, understood. Extra luminal space. So this is the division of it. Okay, so look here, luminal space, and whatever is outside, that is your extra luminal space, basically the cytoplasm. Okay, understood? Yes ma'am, understood. See again, you can see. Now students, I told you, number rough endoplasmic reticulum, that is example is the number endoplasmic reticulum. In the endoplasmic reticulum, சில இடத்துல நம்ம ribosomes இப்படி present ஆயிருக்கும். Ribosomes இப்படி present ஆயிருக்கும். ஓகேவா? இப்படி present ஆயிருக்கும். And wherever that ribosomes are present, அது வந்து நம்ம என்ன கூப்பிடுவோம்? We call it rough endoplasmic reticulum. Rough endoplasmic reticulum. Okay va? Rough endoplasmic reticulum. அதே imagine பண்ணிக்கோங்க students, அதே rough endoplasmic reticulum there will be regions where the protein, the ribosomes will not be there. Okay, this is what, this is smooth endoplasmic reticulum. இது வந்து smooth endoplasmic reticulum. So இதுல main என்ன? இதுல வந்து main என்னன்னா ma'am ribosome present ஆயிருக்கும் ma'am. Ribosome is present. Correct? Yes, correct. Ribosome is present. Okay va? Understood this? Understood? Yes. Okay. Now, look here. The endoplasmic reticulum, okay? The endoplasmic reticulum bearing the ribosomes on their surface, okay? Bearing the ribosomes on their surface is called as rough endoplasmic reticulum or RER. Okay. In the absence of ribosome, they appear smooth. That will be called as smooth endoplasmic reticulum. Okay. In the absence of ribosome, அது வந்து smooth endoplasmic reticulum. Understood, ma'am? Now, smooth endoplasmic reticulum help in synthesis of lipids and hormones. So, நம்ம rough endoplasmic reticulumல நமக்கு ribosome இருக்கு. So, because that ribosome is present, நமக்கு வந்து protein synthesis பண்றதுக்கு ரொம்ப easy. ஆனா இந்த ஸ்மூத் எண்டோபிளாஸ்மிக் ரெட்டிகுலம்ல ரைபோசோம் பிரசன்டே இல்ல. சோ அதனால அது வந்து ஆப்சென்ட். ஓகே, அதனால இந்த ஸ்மூத் எண்டோபிளாஸ்மிக் ரெட்டிகுலம் வந்து லிபிட், ஸ்டிரால்ஸ் அந்த மாதிரி சிந்தசிஸ்க்கு ஹெல்ப் பண்றாங்க. ஓகேவா? சோ ஹியர் யூ கேன் see it is going to synthesis of lipids and hormones. Okay, and RER is frequently observed in the cell, actively involved in protein synthesis and secretion. Why? Because ribosome job என்ன students, function என்ன? Ribosome function வந்து protein synthesis பண்ணணுமா? So அதனால அந்த endoplasmic reticulumல ribosome present ஆயிருந்தா obviously அது வந்து protein synthesis help ஆகும். Yes or no? Yes ma'am, understood. Because ribosome we studied sometime back in translation. அந்த டைம்ல on an mRNA strand multiple ribosomes will be present, so that we call it as polyribosome or polysome. And the ribosome, what it will do, students, with the tRNA, it will read the mRNA and it will make proteins, right? It will attach the amino acid and it will make proteins. If you are a student who hasn't watched Molecular Basis of Inheritance, you can watch that, okay? So, because of that, this is anyway going to do protein synthesis. So, that is very easy for you to understand, right? So, they are extensive and continuous with the outer membrane of the nucleus. So, adhanalada I have attached it with the nucleus. I did not separate it with the nucleus, right? When I am drawing it, I am always drawing it like this. I am drawing the nucleus and I am attaching like this only. So, it is attached to the outer membrane of the nucleus. Outer membrane of the nucleus. Clear? Done. This is understood, all of you. Okay. Next வந்து நம்ம Golgi body. Okay, Golgi body or Golgi apparatus னு சொல்லுவாங்க அது மாதிரி. So Golgi in 1898 first observed densely stained reticular structures near the nucleus. So very close to the nucleus. நம்ம endoplasmic reticulum மாதிரியே தான் கொஞ்சம் கூட அவையான nucleus பாசமே ஒரு Golgi apparatus இருக்கு. So from his name only he gave the name Golgi apparatus. So structures near the nucleus. These were later named as Golgi bodies after him. In plant cell, it is known as dictosomes. Okay, dictosomes, you can pronounce it. These are absent in prokaryotes and they are found only in eukaryotes. This is the exception, important. Okay, okay, what is that? This is not there in prokaryotes, but they are present in eukaryotes. So, number Golgi apparatus, the students. Again, it will look, you can either you can draw it like this, okay, or you can draw like this also. Zigzag Mari, you can draw it like that, okay, like a continuous structure, okay, Golgi apparatus. So it was given by, the name came from Camillo Golgi in 1898. He noticed that there is some structure that is very densely stayed, okay, densely stayed reticular structure which is very, very closer to the nucleus, okay, va? Okay. They consist of many flat, disc-shaped, okay, disc-shaped sacs or cisternae. These are stacked parallel to each other. So just look at the structure, students. So this is the cis phase, this is the cisternae, and this is the trans phase. So cis phase and trans phase, I think you have studied in chemistry as well. We tell cis and trans. Okay, and the like we say like a mirror image type. So here they have a cis phase. Okay, they have the cisternae, which is the very important foldings that we are talking about. Okay, and then we have the trans phase. Okay, trans phase. So we'll learn more about it. So just look at the foldings. Draw pandradhukku. Okay, this is how, like the drawing is little, and I'll draw like this for all of you. Like this only, like this. Foldings. Like this foldings are there. So we have a cis phase, a trans phase, and the cisternae, which is the foldings. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged. Okay, everybody students, concentrically arranged near the nucleus with a distinct convexus or forming phase, or the forming phase, and the concave trans or the maturing phase. Okay, so first is the very initial stage, that is what number cis phase. Okay, and the cis phase we can also call it as the forming phase because this one the initial phase nu sollala. So it's a forming phase. அதுக்கப்புறம் இந்த சைடுல ஸ்டூடண்ட்ஸ் நமக்கு வந்து trans phase இருக்கும். அந்த trans phase வந்து it is the maturing phase. Okay, maturing phase. Okay, understood this much? Yes. Okay, so the Golgi cisternae are concentrically arranged, and இதுதான் நம்ம cisterna. This is the lumen. So எல்லா ஸ்ட்ரக்சருக்குமே அந்த center space வந்து lumen தான். And the corner, so like let's say for every organelle for that matter, and the lumen illana and the space irukum. Okay, and the space irukum. And here students understand, concentrically arranged near the nucleus with a convex. Okay, we have a convex cis phase and a concave trans phase. Okay, I think you have to remember a trick for this. So this is BC, I think I'll put X. Convex, you will remember, concave. Okay, you have to remember XCCT. Okay, XCCT. So convex, cis phase, concave, trans phase. Okay, try to remember something. I don't know what trick, suddenly trick won't come. But then yeah, if I find a trick, definitely I will give it to you in the later. Okay, so don't worry, I'll give this. Okay, so remember convex. Okay, VC. Okay, VC. Okay, VC. So VLC madri, Vedantu learning set, adha madri VC. So at least one you remember, that automatically you remember. So VC, convex, C, cis phase, concave, trans phase, or else it's called the maturing phase, or cis phase is also called the forming phase. Okay. Now the cis and the trans phase of the organelle are entirely different, but they are interconnected. Okay, adu vandu completely different irukka, ana kude they are internally connected. That is, the convex side is known as cis, okay, or else the forming phase, same thing they have given here, and the concave side is known as trans or the maturing phase. maturing phase. They are saying what? They are entirely different. A cis and a trans phase is entirely different. And they are interconnected, obviously, because if you look at the structure, one side trans phase is there and another side cis phase is there. You have the cisterna, loom and everything is there. They're all interconnected. They're all arranged in concentric circles. Okay, so are only interconnected. So look here, students. But these phases have like crazy functions. Okay, these phases have crazy functions. So function patida, it's very important for us to study. Look here. The forming phase receives transport vesicles from the endoplasmic reticulum. The vesicles will fuse with the Golgi bodies and release their content. Okay, so what will happen? One fellow, that is your cis phase Vanda, it is like a receiving phase where it is taking what, students? It receives the vesicles. Inside the vesicles, the materials are going to be there? Yes, the vesicles are going to take it. The vesicles will fuse with what, students? Fuse with the Golgi bodies and they will release their content into it. Okay, so here you can see, it is the anastomosis phase. So here you can see secretory vesicles, they will go and they will add their secretions or they'll put whatever content is there into the Golgi. Now look here, then they pass through the various cisternae and reach the maturing phase. So just imagine it is like, நம்ம வந்து ஸ்கூல்ல அந்த ஸ்கேல் இருக்கும்ல ஸ்டூடெண்ட்ஸ், அந்த பால் இப்படி மூவ் பண்ணி we have to reach to the destination அந்த மாதிரி தான். So vesicle வந்து let's say it started from the எவ்வளவு alarms இருக்கு. Okay, let's say it started from the cis phase, okay, and then it got entered. Now it will move like this through the cisternae all the way down and it will go to the trans phase. Okay, both the sides are there but too difficult to draw both the sides. Okay, like this to the trans phase. So you can remember if like that and the ball marvila, so it has entered, it's moving and now it is in the trans phase. Okay, now then they pass through the various cisternae and they will reach the maturing phase ilena nama trans phase. Okay, clear? Now look here, what is going to happen? Upon reaching the maturing phase, they bud off as secretory vesicles. Okay, pasangla, continue pannalama. Okay, so we understood about cis phase and trans phase. Now let me teach you one additional thing. Okay, not additional, it will make more sense for you when you understand this whole thing. Okay, so look here. Function of Golgi apparatus, what it is going to do, it helps in the formation of the plasma membrane, it helps modification of protein, lipids synthesized by the endoplasmic reticulum. Golgi apparatus is an important site for the formation of glycoproteins and glycolipids. So, this is explain panna poro. Okay, this is nalla gavanikanam students. Okay, look here. இப்போ என்ன நடக்குது, நமக்கு வந்து நியூக்ளியஸ் இருக்கு, correct ma'am. Okay, see, this is our nucleus. Okay, it has double layers, correct? It has double layers. In the double layers we have attachment. Whom do we have attached? We have our endoplasmic reticulum that is getting attached, correct? Yes, correct. Okay, this is our endoplasmic reticulum. Okay, now in the endoplasmic reticulum namakku vandu ribosomes will be attached. Because of that, this will be rough endoplasmic reticulum, correct? Yes ma'am, correct. Adukapram namakku vandu Golgi apparatus will be present. Okay, indha madri Golgi apparatus will be present. Okay, if you haven't subscribed to the channel, make sure that you are subscribing to the channel. Okay, so students, in the ribosome vandu namakku pink color la pannalam. Okay, now listen to me carefully how all these people are connecting. That is why we are calling it endomembrane system. Okay, endomembrane system. So இது வந்து students first நமக்கு nucleus இருக்கு, correct ma'am. அதுக்கப்புறம் endoplasmic reticulum இருக்கு. இந்த endoplasmic reticulumல நம்ம rough endoplasmic reticulum வந்து it is going to help for protein synthesis. Yes, it is going to help for protein synthesis because I already made you understand that நமக்கு ribosome இருக்கு, so அதனால protein synthesis வந்து நடக்கும். ஆனா நம்ம smooth endoplasmic reticulum வந்து it is going to help us in the lipid and hormone formation. So lipid formation க்கு help ஆகும். இப்போ என்ன நடக்குது students, இந்த protein synthesis வந்து it is going to be, that is the proteins are going to be picked up by your cis phase, and the cis phase, what do you call it as? It is also called as the forming phase, right? So look here, I will just write here protein that is formed, it is going to be picked up by your forming phase, okay, forming phase. Okay, in the forming phase la nama cis phase irukku, cis phase is also called as nama forming phase, okay, and then it is going to be passed on with the help of your cisternae, it is going to be passed on and it is going to come ultimately where it is going to come to the trans phase or else the maturing phase. So, that protein has moved and it has now reached the trans phase, illena it is also called as nama maturing phase, okay, maturing phase. After this maturing phase, students, these people are going to help. From there, it can go to different places, okay. So, it will reach the maturing phase and it can become right now a glycoprotein. It can form what? A glycoprotein. GP I am writing. So, that is a glycoprotein. Correct தானே? Yes ma'am, correct. ஒரு glycoprotein it is forming. அதே மாதிரி தான் நம்ம lipidsum வந்து same thing, okay. It is going to be taken by the forming phase. It is going to be moving all the way. It will move through the cisternae and it will reach the maturing phase. From there, it is going to help in the formation of the GL, that is glycolipid. Glycolipid. So, this is how the entire system is going to work, right? So, how students this entire system is going to work? நமக்கு வந்து nucleus இருக்கு. From the nucleus நமக்கு வந்து endoplasmic reticulum is connected. அந்த endoplasmic reticulumல RER இருக்கு, SCR இருக்கு. RER வந்து it is helping us in protein formation, that is protein synthesis, okay, and SCR வந்து நமக்கு lipid formation or lipid synthesis, and this both are going to be taken up by the cis phase or the forming phase, and from there it is going to be transported with the help of cisternae. It will move through the different cisternae, it will reach the trans phase. From the trans phase, it is going to form a GP and it is going to form a GL. Okay, va? Okay, this is the kind of clarity that you have to have. That is when you will understand why did we call this the endomembrane system. Okay, endomembrane system. Understood? They have connections, they are helping each other. Okay, idhukku appuram students namakku vanda it also helps in cell wall formation or a new cell wall. Okay, Golgi வந்து lot of functions are there. Next வந்து நமக்கு வந்து lysosomes. Okay, lysosomes. Lysosomes were discovered by Christian D. Douve in 1955 and he noticed what? He noticed a lysosome. Lysis na enna digestion. Lysis na enna digestion. Some na enna body. Okay, when I am saying Golgi body, mitochondria, ellame body da. Some word varudhu body. So, soma means body. These are small vesicles which are bounded by single membrane. Okay, it is bounded by single membrane. நமக்கு வந்து plasmid double membrane, நம்ம nucleus வந்து double membrane, okay, mitochondria வந்து double membrane, ஆனா இது எல்லாமே single membrane structures, okay, single membrane. So, that is your lysosomes. Now, what does this mean? These are membrane bound vesicles, okay, another membrane bound vesicular structures formed by the process of packaging in Golgi apparatus. So, it is one way I can say that it is formed by the Golgi itself. In the lysosome one, it is actually formed by the, we say Golgian vacuole. Golgian. Okay, it is formed by the Golgian vacuole, we can say. So, it is a, like we can say it's like a byproduct or like a product of our Golgi apparatus, this lysosome. Okay. So, inga paakumbodhu see students, you can see protein vanda it is going to the transport vesicle, getting into the Golgi apparatus, reaching the trans phase in a, it is going to different places and then it forms the lysosome. So, and the madri vesicles will be formed and then finally it is forming it, formed by the process of packaging of your Golgi apparatus. Lysosome is a very rich in almost all the type of hydrolytic enzymes. That means it will be like it is ready to digest them, but it will give a lot of water and digest them. And the madri, okay, like I am giving you a lot of water and then I am killing you. And the madri. So, and the madri da hydrolytic enzyme na in the hydrolases, that is what and all lipases. Lipases we have studied, yes ma'am, to degrade lipid, then proteases to degrade protein, carbohydrates to degrade carbohydrate. Ana idu ellame hydrolytic, okay, hydrolases. Water kuduthu adukapram, okay, it will finish the story. So, the enzyme functions in a acidic pH of 4 to 5. Students, if I have to make the lysosome or work then I have to give it a acidic pH. So then from where I am going to give the acidic pH? I am going to give it from the cytoplasm. So this is our lysosome. Okay. In the lysosome, proton is necessary because it has to become acidic. So I am going to give the protons. Okay, I am going to give the protons with the help that is from the cytoplasm. So Anna, in the lysosome already rich concentration or high concentration of proton irkume. Yes ma'am, then only it is going to work. So on the concentration, how will I move it? So for that I will have to use a pump. Yesterday only like when we studied the neural control, day before yesterday when we studied neural control, I told you pump is necessary. That means energy is necessary if I have to move something from low to high. So adanale here also we will be using pump. We can use a proton pump. Enna pump? Proton pump to make this lysosome acidic so that they can work happily. So they are known as acidic hydrolysis. Okay, is this clear? So this is acidic pananame. So adanale na from the cytoplasm na hydrogen I am pumping it inside the lysosome so they can work properly. For that pumping also I will need a proton gradient or I will need a proton pump so that the hydrogen can be moved. Okay. Now these enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acid. So basically they are like a suicide bag. We say lysosomes as the suicide bag of the cell, where it is going to digest all these people who are very very necessary, but when it's they know, the cell knows that when it is necessary, when it is not necessary. So we can consider it as like a suicide bag. Okay, clear to all of you? Okay, hydrolysis of what and all? Carbohydrate, protease, lipases and your nucleases. Okay, all this is going to be there. Understood? Screen is not visible. Huh? Visible, no? Okay, understood ma'am. So this notes will help you study faster. Okay, look here. So it digests excess or worn out of organelles and engulfs the virus or bacteria. So they keep the cell clean. Okay, they keep the cell clean. Whenever something has got worn out, something is excess, adellame idu vandu digest pandranga. So adanal da carbohydrates, lipases, proteases and nucleases ellame present aayirukku. Autolysis, it refers to self destruction of a cell, that is it occurs in aging. Dead or deceased cell also will be. Okay. Now next topic on the students nama vacuole. So in membrane system we finished endoplasmic reticulum, we finished Golgi apparatus, we finished lysosome. Lysosome only that much is there, that they have hydrolytic enzyme, that means they have a lot of water and then they will digest all these people. They will digest what and all? Nucleic acid, carbohydrates, lipids, proteins, idellame digest pannum. When do they digest it? When it is excess or when it is worn out, when it is old, when it is worn out, idu vandu digest pandranga. Not just that, or bacteria and a virus also when it enters, it can engulf it, it can do phagocytosis and it can digest it. So avloda lysosome. Ana in the lysosome it is like a product of nama Golgi apparatus. Golgi apparatus packaging kapra maybe like a byproduct madri or Golgian vacuole will be used to make this lysosome. Okay, lysosome. So next vandu namakku vacuole. Vacuole la enna, it's like a storehouse. Okay, nama veetla ellame oru cupboard irukum illana nama room la vandu oru chair irukum where every clothes in the planet will be on that chair. Okay, from bed we will put it on the chair, from chair we will put it on the bed. Okay, if I need the chair, I will take it from there and throw it there. And the madri, or a storehouse this vacuole is. Vacuole has everything that the cell doesn't want. So, students just imagine panikonga, this is your room. Okay. This is your room. Okay. You have kept the room all clean. But Anna, you have that one chair or one cupboard which looks like if someone opens it, they are going to faint. So adha nama vacuole. Vacuole la vandu ellame store agum because so that the cytoplasm can be very very organized. Cytoplasm vandu it is very organized. So adanale whatever is necessary, unnecessary ellame it will be dumped into the vacuole. So vacuole will be already overloaded with stuff. Okay, overloaded with stuff. Look here, the vacuole is a membrane bound space found in the cytoplasm. Vacuole contains water, sap, excretory product and other materials not useful for the cell. Okay, the vacuole is bound by a single membrane called as tonoplast. So in the membrane that covers the vacuole, we call that what? Tonoplast. It's important. Okay, it is single membrane structure, so we call that single membrane as what? tonoplast. We have different type of vacuole, right? In smaller classes also we have studied contractile vacuole, sap vacuole, so and the food vacuole. So there are different types of vacuole who is going to do the functions. One is going to help in excretion, one is going to digest the food, and the different types of vacuoles will be there. And generally vacuole meaning enna nama storehouse of the cell nu sollala. Wherever unnecessary necessary ellame nama vacuole la dump pannita cytoplasm vandu it will be like, oh, I want to be organized. When someone comes nama hall madri, nama hall vandu super duper clean ah irukum, ana rooms ellame கச்சடா மாதிரி இருக்கும். அந்த மாதிரி தான் நம்ம சைட்டோபிளாசம் வந்து ரொம்ப ஆர்கனைஸ்டாக இருக்கும். ஆனால் வேக்யூல் வந்து okay gone. So look here, in plants the tonoplast facilitates the transport of, okay, what does it facilitate? The transport of a number of ions and other materials against the concentration gradient into the vacuole. Hence their concentration is significantly higher in vacuole than the cytoplasm. So normally students, nama cytoplasm and it is very, it's like I want to always keep it clean. So adanale what the cytoplasm will do, cytoplasm even in the smallest like problems, okay, like let's say something is unnecessary, cytoplasm can keep it. Ana kuda in the cytoplasm and it will try to dump it in the vacuole. ஆனால் vacuole already higher concentration இருக்கு. Okay, vacuole is already in higher concentration and the cytoplasm obviously will not have that much of useless substance. So still it will try to push it. Still it will try to push everything. Even though vacuole will have a higher concentration, cytoplasm வந்து இன்னுமே அது force பண்ணி you take it எனக்கு வந்து clean-ஆ இருக்கணும், so அதனால you take it மாதிரி it will push it. Okay, it will push so much like that you only take it. Clear? Okay, clear? Yes, plant cell will have bigger vacuoles, animal cell will have smaller vacuoles. Plant cell are going to have, like the vacuole size is going to be very big, okay. They store excretory material, they store everything and keep, okay. Done. Now look here, depending on the content and the function, vacuoles are of different types, okay. Vacuoles are of different types. Namakku vandu sap vacuole irukku, where the sap will be stored. Adukapra air vacuole irukku, contractile vacuole irukku, food vacuole irukku. This contractile vacuole and food vacuole vandu nama amoeba, when we were studying amoeba in your life process chapter 10 standard la, we have studied about contractile vacuole and food vacuole, if all of you remember, okay. Sap vacuole enna irukum students? Obviously ma'am, sap irukum ma'am. It's a fluid known as the cell sap or the vacuolar sap. It occurs in animal cell and also in young plant cell. Contractile vacuole helps in osmoregulation. Osmoregulation ke help pandra vacuole, vacuole vanda obviously it is going to help for excretion as well. So this is going to help us in excretion, not as like in small organism, in excretion. Example vanda nama amoeba. Okay, amoeba la irukku contractile vacuole. So sap vacuole la vandu sap irukum, contractile vacuole la vandu excretory function irukum, and with that they are also going to do osmoregulation. Now food vacuole will fuse with the lysosome. Lysosome have digestive enzymes, okay, which digest the content of the food vacuole. So the digested food is absorbed, the undigested food will be thrown away. Again in amoeba only we study food vacuole. Food vacuole enna nadakum students, if it comes in contact with the food, okay, if it comes in contact with the food, just imagine here food is there, okay. If it comes in contact, it will try engulfing, okay, it will try engulfing, and what will happen, idu vandu engulf ஆகி, okay, இந்த மாதிரி engulf ஆகும் and it will get inside like this, okay. It will start engulfing and then it will enter inside and it will get digested and it is going to get attached with lysosomes. Why so? Because it will fuse with lysosomes, because lysosomes have digestive enzymes, okay. Digestive enzymes on the present ஆயிருக்கு. So அதனால இந்த lysosomes will help in the digestion of the food, and the waste material can be sent out through the contractile vacuole, okay. So digestive enzymes are present in lysosome which will help in the digestion, okay va. Now air vacuole store gases and provides buoyancy, okay. They will store gases and provide buoyancy. Idu ellame don't have to break your head for, but then endoplasmic reticulum, Golgi apparatus, idu ellame important. So idu ellame you have to study properly, okay. trances and all of that. Next வந்து நம்ம powerhouse of the cell. எல்லா students-உமே, like let's say art student, math student, LKG student also know that mitochondria is the powerhouse of the cell. Why? Because they help in ATP production. So in the mitochondria patti we are going to study more and more about mitochondria today. So look here, mitochondria is generally found in aerobic eukaryotes. Aerobic na enna? The ones that will require oxygen. Okay. Now, absent in prokaryotes and anaerobic eukaryotes, cells of germinating seeds have greater number of mitochondria than a dormant seed. Okay, the cell that is doing some job will have more mitochondria because it needs energy. Okay, why will a cell who is sleeping like this will need mitochondria? It does not require mitochondria. Now, mitos means enna students? Thread. Okay, chorion means grain. Okay. So, they were the first observed by Colliker in 1880. Benda named them as mitochondria. So, one person found it, another person named it. Okay, another person named it. Now, they have a length of 1.1 to 4 μm. Okay, and the diameter is about 0.2 to 1 μm. Okay. Avloda very small in size compared to chloroplast and all. It is very, very small in size. Chloroplast and all is 3x bigger than this. Okay, 3x bigger than this. So, this is the size. Now, they have two membranes. So, this is the double membranous structure. This has two membranes, inner and outer membrane. Each membrane. So, nama membrane draw பண்ணும்போது students, நமக்கு வந்து see like this you can draw. Okay. I am writing and drawing this side only. So, you don't have to. I have drawn this. Remember in respiration when we studied, we have already studied all of this. Okay, dark reaction, etc. So, okay, like this. So, this is the structure. So, this is the outer membrane, this is the inner membrane. Okay, wow. Each membrane is made up of a lipid bilayer. The space between the two membranes of the mitochondria is called as the perimitochondrial space or the intermembrane space or the outer chambers. So, look here, this is the inner membrane. Okay, inner. This is less permeable. Okay, less permeable. Okay, adukaprom yellow color la draw pandren. This is the outer membrane. This is obviously students more permeable. Okay, this is less permeable aayirunda, this is more permeable. That is like it's like so simple to remember. Okay, this portion where all of this is there, no, this is nama matrix. Okay, this is nama matrix. This is nama perimitochondrial space. Perimitochondrial space. Remember perimitochondrial space again in ETS we have studied. Okay, now look here. The inner chamber contains of semi-fluid matrix. Inner chamber la enna irukku? Semi-fluid matrix. That's what I labeled. Matrix has protein particles, ribosome, RNA, DNA, enzymes of the Krebs cycle. Outer membrane is smooth and permeable to large number of metabolites due to the presence of porins. Okay, avarku enna irukku students, there are structures which are like small pores are there, adanal adu vandu more permeable irukku compared to our inner membrane. Okay, compared to our inner membrane. Major mitochondrial activities takes place in the inner membrane. To increase its efficiency, it is infolded to form cristae. Okay, cristae. The density of cristae indicates the intensity of respiration. Density of cristae indicates what? Intensity of nama respiration. This draw pannalama? Okay, idukapra. Okay. Inner membrane as well as the cristae have elementary particles as like F0 or F0 and F1 particle, and the proton gradient nadakkiradhukku F0, F0 and F1 particles are there. Because we need Tamil Nadu students have already studied respiration, இது எல்லாமே to understand it is very very easy. These have head, stalk and base. நமக்கு already தெரியும் நம்ம F1 particle-க்கு ஒரு head and stalk இருக்கு, அதுக்கப்புறம் F0 வந்து ஒரு base மாதிரி இருக்கும். So head and stalk is going to be present and base is going to be present. Okay. Okay. This elementary particle functions as an ATPase. This also we know. Yes, ma'am, we know this also, that we have this one working as an ATPase, and they are centers of ATP during oxidative phosphorylation. They are centers of ATP during oxidative phosphorylation. After this, if you want to once again go and watch respiration, you can go and watch respiration, because you will understand more. Okay, you will understand more. Yes. Now, the matrix also possesses few RNA molecules, ribosome and components required for the synthesis of protein. So, I will draw it here, students. After that, again I will explain it to you. So, it will become very easy for you to understand. Okay, yes. So, this is our mitochondria. Inge ma'am, mitochondria. Okay. Students, it's a semi-autonomously working cell. Semi-autonomous na enna? It is not as like, it cannot work like a cell completely independent. Ana kuda semi-autonomously it can work, because adila ribosome irukka, RNA materials irukka, protein irukka. So adinala mitochondria can function. Okay. Now look here, it is a double membranous structure. So here you can see. Okay. Okay. இந்த மாதிரி foldings இருக்கும். இந்த foldings we call it as cisternae. Sorry, cristae. Cisternae is in your Golgi. Okay, like this foldings will be there. Okay, like this foldings will be there. These foldings are there. Now if you look at it, students, this attachment is not proper. Okay. So, look here, this is our outer membrane, correct ma'am, this is our outer membrane. In the outer membrane, enna irukku students, porins are present, enna irukku, porins are present. So, here I will just draw like, you know, okay, like this structures. So, these are your porins. Because this is present, idu vanda more permeable ah irukku, okay. This is permeability is more. Arrow mark when I am indicating to the top, that means it is more permeable. Okay. When I am indicating like this, it means more permeable. Okay va. So, this is more permeable. Adukapra idu da namma inner membrane. Okay, idu da namma inner membrane. Inner membrane la da students almost everything is happening, because inner membrane is more you know, efficient inner membrane. Idula vanda less permeable. Idula vanda less permeable. Okay, less permeable ah irukum. Adukapra in the structure illa students, this is the matrix. Okay, this is the matrix. Okay, and this foldings are what? Cristae. In the foldings the cristae. Okay, this infoldings are necessary for them. Okay, so they can, you know, to increase the surface area. So, idhukku ellame function irukku, it is to increase the surface area. Again I am showing arrow mark to the top. Okay, and also remember these have your F0F1 particle. Okay, F0F1 particle. In the F0F1 particle enna nadakum, they will have a head. Okay, they will have a head. Okay, then they will have structures like this. Okay, okay, they will have structures like this. So, the movement of electrons can happen and protons can happen, so that the energy formation will happen. Okay, they will have ATPase activity, this we already know. Okay, ATP activity will be there, so the movement can happen. Okay, done. This is understood? Yes ma'am, understood. Now I told you students, in the cell one the semi-autonomous, and it means it has almost everything that, you know, is required for them to successfully live their life. So adanala they will have the okay, like this structure will be there. Then they will have what? They will have some RNAs as well. Okay, they will also have some RNA. Okay, RNA like this will be there, single stranded RNA will be there. With RNA they will also have you know, They will also have 70s ribosomes, so they can make their own proteins, right? They will have 70s ribosomes, own proteins can be made. So they are very self-sufficient. They are like, okay, I can do most of the things by myself. So adanale we call them semi, not fully, semi-autonomous. Okay, we call them what? Semi-autonomous. Okay, va? Yes, ma'am. Okay. Okay, okay. Yes. Understood, right? Yes. Okay. This is your double stranded structure. This you already know, double stranded structure. Now, yes, students, in the mitochondria one, it will be like a sausage shape. Okay. It will be like a sausage shaped structure. Sausage shaped structure. Okay, sausage shaped structure. Like if you look at it, no, it will have a sausage shaped structure. Is this clear? Yes, ma'am, it is clear. All of you? Yes, I hope everyone are understanding everything. Okay. Now students, in the cell one, it's very small, right? It's very, very small. So adanale we will have to use a dye which is called as Janus green. Okay, we have to use Janus green dye to actually visibly see this mitochondria, because it is so tiny. It is so tiny. In the dye one, it is special. So if you dye this in the cell, you will be able to see mitochondria. Elena, it is not visible. So it is not visible because it's very small. So in the dye da use panni namakku vanda mitochondria visible ah theriyum. Okay, visible ah theriyum. So this is understood. Yes or no? Mitochondria is done and dusted. So this much we have understood. Again, we have to study some more things like, see, DNA can replicate independently. DNA can replicate independently and can produce their own mRNA, tRNA and rRNA. DNA is naked and it is circular or it can be linear. So mitochondrial DNA on the circular types, it will be double stranded structure. Okay. Now new mitochondria can develop by binary fission of the pre-existing mitochondria. This might be new information for all of you. So adanale just make a note of this. Okay. Now we are going to the next topic, that is your plastids. Before that, let's just drink some water. Okay. I'll label this also so that you know, peri, this also a label. So students, in the blue color structure, this one, okay, like this, okay, like this, okay. I am just drawing few of them for you to identify that, okay, these structures are present here. Now here I will also label the perimitochondrial space. Perimitochondrial space. Okay, perimitochondrial space. So we have labeled the entire structure. Okay, next one the students, nama plastids. Yenna ma'am, in the plastids. Okay, plastids. So look here, the term plastid was introduced by Haeckel. Okay, they are restricted to plant cell only. They are present only where? In the plant cell. Depending on the color, plastids are 3 types. Depending on what? Depending on the color. So in the word one important, depending on the color. First one nama chloroplast. Chloroplast namak already theriyume, chlorophyll will be present. Then we have chromoplast, which will have other colors like your xanthophyll, keratin, nama carrot la illana nama green plants la nama crush pannita paper chromatography பண்ணும்போது we will see slight one orange color, slight one yellow color. அது எல்லாமே because of other pigments as well, not just chlorophyll, right? And then we have leucoplast, which is more of whitish color, like colorless it is. Okay, now look here students, leucoplast, they are colorless plastid. Another colorless plastid. So how will you remember? Our leukocytes, nama WBC also is colorless. So adanale you can relate to it, telling that, okay, leucoplast na it is going to be colorless. Okay, leucoplast na it is going to be colorless, which generally occur near the nucleus in a non-green cell. Obviously it is colorless, it is not going to be present in green cell. Again, adukku vanda classification irukku students. amyloplast. Amyloplast na enna? Amylase. Amylase word verumle namakku already theriyum. Adu vandu starch related ana oru word. So amyloplast na starch storing plastids. So in the structure vandu plant cell la special ah present aayirukkum. They are going to help in storage of one or the other thing. Okay. Adukku next one we have alloplast. Alloplast is going to help in oil and fat storing plastids. Enna oil adukapram fat storing. Okay. Then we have aileroplast. Aileroplast வந்து protein storing plastids. நம்ம aileron layer. Okay, aileron layer மாதிரி தான் protein. Okay, protein. So it is going to be protein storing plastids. So amyloplast, alloplast and everything is coming allo. Okay, aileroplast. Aileroplast வந்து protein. Then we have oil and fat. அதுக்கப்புறம் நமக்கு வந்து amylase, that is your starch storing plastids are going to be there. In the leucoplast it is colorless. Okay, this is something that you have to remember. Based on color, this classification has been done. Ana idu vanda colorless plastids. So remember leukocytes. Nama WBC vanda colorless irukka. So adanal nama leukoplastum colorless irukkalam. So andha madri you should make connection between all the topics. Illana it becomes difficult. Chromoplast students, they are yellow or red in color and provide color to fruits and flowers. Okay, so in the chromoplast is like a more colorful person among all the three of them. One is like I will only do green, yaar, I only like green. One is like yaar, I am a white person, I will just do white. Not that white person, white, I only like white. Then we have chromoplast who is more colorful. Okay, he has yellow, red, so andha madri colors. Change of color from green to reddish during ripening is due to the transformation of chloroplast to chromoplast. Okay, so on the plastid one the change is allowing them to go from green to red. Okay, green to red. So now here we have to study chloroplast in detail. Chloroplast again we have already studied in photos, and that is dark reaction, light reaction also, but Anna we have to study it here once again. So they are green in color. We already know chloroplast is green in color. It possesses photosynthetic pigments. Okay, photosynthetic pigments, that is your chlorophyll and carotenoids. Okay, which take part in the synthesis of the food. It is covered by a double layered membrane. So idum double layered membrane da. Each membrane is 90 to 100 Armstrong. Okay, now two membranes are separated by 100 to 200. Okay, 100 to 200. So you can just imagine students, idu vandu mitochondria madri compare பண்ணும்போது it is 3x bigger than mitochondria is very very small, right? Mitochondria was very very small, ana idu vandu double triple bigger it is. So the ground substance of the chloroplast is known as the matrix, or it is known as the stroma semi-fluid structure. Okay, where are you seeing the stroma? This is the stroma lamellae. This is your, again we will come to this, granum, thylakoids and all of that. Now the DNA of the chloroplast is naked, circular or linear. Chloroplast also has its own ribosome, that is around 70S ribosome. With the help of the ribosome, the chloroplast is able to synthesize most of the enzymes required by it. In green algae, the chloroplast has pyrenoids to store the starch. Okay, chloroplast has additional structures to store the starch. Now flattened sac-like structure run throughout the matrix, which is known as stroma thylakoids. Nama coin madri students, ipo nama vandu, let's say I am keeping coins like this. Okay, or I am like stocking anything like books or something like that. If I stock like this, எப்படி இருக்கும்? Yes ma'am, it will look like one stocking. Okay, and the stocking madri da in the grana also will be present. So the stroma thylakoids are stacked at a place to form the grana. So idu vandu grana. Okay, idu vandu grana. Okay, then the stroma thylakoids will be stacked at a place to form grana. Each can go up to 2 to 100 thylakoids. Grana are absent in algae chloroplast, so they are known as agranule. Okay, agranule, this also I have taught you. Ana single structure ku enna koopuduvanga? Thylakoids nu koopuduvanga. Okay, thylakoids. If a students, one grana has to be attached to another grana, yes or no? Yes ma'am, one has to be attached to another. So for the attachment, there are going to be structures which are your lamellae, or we can also call it fret. Okay, we can call it what? Fret. Stroma, lamellae or fret. Okay, you will be telling ma'am, you please draw ma'am. then only we will understand. I know most of you will be telling that. Okay. Double membranous structure. Okay, yeah, almost okay, yeah. So, in the structure la enna important na, in the thylakoids arrangement da important. Okay, in the madri, coin madri, it will be attached like this. Okay, like this it will be attached. Again here also many will be there. Each of them can have 2 to 100 thylakoids. Okay, like this it will be attached. And wait, to connect from one thylakoid to another one or grana to another one, or stroma lamellae will be present. Enna irukku, or stroma lamellae will be present. So, labeling pannalama. So, in the mic one, the students, it's like singing song for me and rotating around itself. So, idu da nama outer membrane. Okay, double membrane na, or outer membrane irukku, or inner membrane irukkum. So, this is our outer membrane. Okay, idu da nama inner membrane. Okay, idu da nama grana. Okay, grana. Idu enna single one vandu nama thylakoid. In the attachment ku nama vandu stroma lamellae. Okay, stroma lamellae. So, after this, once you please revise photosynthesis and your respiration chapter. Okay, many of you would have forgotten so many things, that is why. Okay, between the students there is intermembranous space. Intermembranous space irukum. This also we have studied. Remember IMS or IMM, intermembranous matrix. Remember you have studied this also while studying our electrotransport. So, yes, and students, they will have their own circular circular structure. Adukapram they will also have their own ribosome, so they can synthesize their own proteins. Okay, 70S ribosome. Correct? Yes, ma'am, correct. Okay, that's our double stranded circular DNA. Circular DNA. And students, this is our stroma. Okay. So, in stroma what will happen? Dark reaction will happen. All of you remember? Dark reaction will happen in the stroma. Dark reaction. So, in the grana what will happen? Light reaction will happen. Avaloda. Okay, this is understood? Yes. Dark reaction will happen in the stroma. Light reaction on the membrane of the thylakoid. So, here I am writing light reaction will happen in the membrane of the thylakoids. This much is understood? Okay, this was easy, right? Yes or no? I am fighting with the mic. Okay, yes ma'am, this is understood. Okay, now we will come back here. Almost, yes. Okay, here I will write about the thylakoid number also. So, one grana can have around 2 to 100 thylakoids, and students, we do not have to stain. Okay, no need to stain this. Okay, no stain is required for seeing this. Okay, to see a chloroplast you do not need a stain, like how you stain the mitochondria with Janus green, and the madri no stain required. Okay, no stain is required. Okay va, avaloda, no stain is required. Clear? Yes ma'am, clear. Now we have to study once again about ribosome. Ribosome we already studied the main part, but little about discovery we have to study. So, adanale in the ribosome vandu inga add aachu. So, George Plastids in 1953 observed dense particles under the electron microscope. Who? George Plastids in 1953. So, he observed some dense particle under the electron microscope. These particles are known as ribosome. Ribosomes can be seen scattered in the cytoplasm. This already theriyum ma'am. They are spherical in outline, covering membrane is absent. It is made up of two subunits, which is known as your small and large subunits. Correct? Yes ma'am, small and large subunit நமக்கு already தெரியும். We know in prokaryotes நமக்கு வந்து 70S present இருக்கும். ஆனால் அந்த 70Sல இந்த S வந்து நம்ம sedimentation factor இல்லைன்னா Sweatworks unit. ஸோ அதனால் இந்த 70Sல larger subunit வந்து 50S இருக்கும் and smaller subunit வந்து 30S இருக்கும். அதே மாதிரி நம்ம eukaryoteஸ்ல 80S இருக்கும் and 80Sல larger subunit வந்து 60S இருக்கும். அதுக்கப்புறம் 40S வந்து நம்ம smaller subunit இருக்கும். இந்த both the units are going to help for protein synthesis. இது வந்து already we have detailedly done. ஆனால் only about George palette we had to study. So அதனால that is there. The size of the ribosome in prokaryotes 70 and 80. S stands for sedimentation. They are composed of ribonucleic acid, that is your RNA and the protein. So in the ribosome வந்து it is mainly made up of நம்ம proteins as well as நம்ம RNA. Ribosomes are popularly known as protein factory. Why? Because they are going to help for protein synthesis. So here you can see protein factory of the cell converts DNA to RNA, converts RNA to protein, modifies protein, packaging of protein. இதெல்லாமே capable of ribosomes are capable. Here you can see நம்ம nucleus வந்து it is going to help for DNA to RNA. We already understood. Yes ma'am, this we already understood because நம்ம transcription வந்து it happens in the nucleus. அதுக்கப்புறம் translation வந்து it happens in the cytoplasm. So அதனால ribosome will form the protein. So அதனால தான் நம்ம வந்து அந்த mRNA we are sending to the nucleus telling you go there, like you go out of the nucleus because cytoplasm la da nama ribosome present ஆயிருக்க. அதுக்கப்புறம் நம்ம endoplasmic reticulum நம்ம protein modification வந்து endoplasmic reticulum will do. எதுவுமே package பண்ணனம்னா நம்ம வந்து automatically Golgi apparatus க்கு we will be sending it for packaging. Okay, clear? Yes ma'am. Now students, in the starting I had told you starting of this that endomembrane system நான் வந்து micro bodies பத்தி பேசி but I didn't tell you a lot about it. So here we are going to study a little about micro bodies, some other small structures which are present in the cell which are going to help in converting some toxic substance to like what is that, non-toxic substance like your peroxide and the madri. So here you can see micro bodies are small cell organelles which are bounded by a single membrane. They are present both in plants as well as in animal cells. They absorb molecular oxygen. என்ன பண்றாங்க? Absorb the molecular oxygen and take part in oxidation. Two of them we have perioxosomes, okay, and glycosomes, okay, glycosomes and peroxisomes. So in the peroxisomes are covered by a single lipid bilayer and they contain oxidative enzymes like a urate oxidase. If you cannot remember this, it is okay, but understand that they have oxidation nature. After oxidation, they release hydrogen peroxide which is metabolized by the cell. What will they do after oxidation? They will release this hydrogen peroxide. Anna in the micro bodies I told you, they have to do the conversion, okay, they have to convert it. They cannot keep the hydrogen peroxide because hydrogen peroxide is toxic for the cell, okay. They have to convert it to H2O plus O2 and this O2 will be utilized by the cell. So glycosomes are microbodies which contain enzymes for oxidation of fatty acid, okay, oxidation of fatty acid. Avloda students, you don't have to break your head a lot for that, okay. Cytoskeleton also I had taught you in the starting that we have microfilaments, microtubule. I told you later when I ask you question you should be able to answer it. That is what this is. An elaborate network of filamentous proteinaceous structures present in the cytoplasm is collectively called as cytoskeleton. Cyto na cell, skeleton na framework, and the framework வந்து யார் provide பண்றாங்க? அந்த microfilament, microtubule, intermediate filament இதெல்லாமே form பண்றாங்க. So the cytoskeleton in a cell are involved in many functions such as what? Mechanical support. I told you just like நம்ம skeletal system மாதிரி தான் they are going to help. Motility, remember microtubule வந்து tubulin protein flagella, so motility, okay. அதுக்கப்புறம் maintenance of the shape. So here you have to remember what I taught you that time. Microfilament, okay, microtubule, அதுக்கப்புறம் intermediate filament. இதெல்லாமே நம்ம cytoskeleton structure, okay, to provide the proper shape, maintenance, motility for the cell. Because our eukaryotic cell is rich in cytoskeleton and locomotory structures. So அதனால these three are going to help us for that. Cytoskeletons are going to be there. Now coming to a very difficult for students concept, cilia and flagella. So before that let me drink some water, okay. Make sure to smash the like button. Okay. Cilia are small structures which work like oars. Oars na enna students நம்ம boatல போகும்போது அந்த both the sides நம்ம oars use பண்ணும்ல இதுதான் oars. Okay, இந்த மாதிரி oars. So we are using that oars, no? அது வந்து என்ன பண்றாங்க automatically it is pushing the water, at the same time it is pushing the boat also in the forward. So அந்த மாதிரி தான் cilia will it will cause the movement of neither the cell or the surrounding fluid. Correct. நம்ம boat move பண்றாங்க, with that the fluid also is moving. So neither the water will move or the boat will move. So அந்த மாதிரி தான். So it will cause the movement of neither the cell or the surrounding fluid. Both are the same because if both are moving, obviously one of the things has to happen. Flagella are comparatively longer and responsible for cell movement. So this chapter you will notice many things are again detailedly explained after already talking about it. So this chapter, that's why most of the students find it difficult to read it from NCERT. So one shots will help you. Okay, இதுதான் நம்ம flagella. So இந்த flagella ல நமக்கு வந்து already ஒரு basal body இருக்கு. Okay, basal body is present. அதுக்கப்புறம் hook is there. Okay, and the longest structure which is நம்ம filament. So in the flagella students, flagella, cilia எல்லாமே we are going to study in cross section. Okay, ரொம்ப brightness இருக்கு. Cross section we are going to study. Clear? Cross section you have to study, so that cross section only will help you understand this whole structure. So let's understand. They are covered. So here we are going to study about the cilium or the flagellum. If I cut the structure, how they are going to look like. So look here, they are covered by plasma membrane. Their core called as axonema. Axonym. Okay, axonym. In the structure students, this is your axonym. Okay, axonym is this structure. So if I have this, let's say this is the structure. Okay, in the structure inge cut பண்ணும்போது I am going to get a circular structure. Yes ma'am, you are going to get a circular structure. And the circular structure தான் entire structure நம்ம வந்து axonym னு சொல்லுவாங்க. Okay, என்ன சொல்லுவாங்க? Axonym. I think I can't reduce any more brightness, so you people have to adjust a little. Okay, so axonym possesses a number of microtubules running parallel to the long axis. Okay, parallel to the long axis. So you can see microtubules are there this way, and then here also you can see idu vanda 9:0 that I'll teach you. So we can call this duplets. Okay, இது எல்லாமே duplets தான். This is a duplet, 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 this is a duplet. So பார்க்கும்போது 9 duplets are going to be there. இது வந்து singlet. Okay, single இருக்கிறாங்க. இது எல்லாமே duplet. Okay, the axonym usually has 9 pairs of duplets of radially arranged peripheral microtubules and a pair of centrally located microtubules. So இதுதான் நம்ம centrally located microtubules, இதுதான் நம்ம peripherally attached microtubules. So students normally பார்க்கும்போது இது வந்து easy தானே. See, இதுதான் நம்ம structure. இந்த structureல இங்க வந்து நமக்கு duplets இருக்கு. எவ்வளோ இருக்கு? 9 இருக்கு. Okay, how many are there? 9 are there. And then in the center நமக்கு வந்து singlets are there. Okay, center la vandu singlets are there. What are all of these structures? Nama microtubules. All of these are microtubules because motility ku help pananamna obviously microtubules da present aayirukna. So adanal these are microtubules. Okay, peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of the axonymal microtubules is referred to as 9+2 array. 9+2 array na enna? இது எல்லாமே duplet தான். அதுக்கப்புறம் 2 singlets are going to be present. So it is going to be 9+2. Correct? Yes or no? 9+2 is understood, no? 9 microtubules in the peripheral, in the center they have 2. Okay? So இது பார்க்கும்போது students it looks like a cart wheel. நம்ம bullock cartல எல்லாமே இருக்குல்ல cart wheel. இப்படி rings இருக்கும். So அந்த மாதிரி இருக்கு. Okay? So arrangement of your axonymal microtubules are referred to as. So what happened right now? நம்ம வந்து அந்த flagella இல்ல cilia நம்ம இப்படி cut பண்ணிட்டு there is a circular structure, and the circular structure we call that as the axonym. அந்த ஆக்சோனியமில் நமக்கு வந்து பெரிஃபரல் ஸ்ட்ரக்சர், பெரிஃபரியில் நமக்கு வந்து டியூப்லெட்ஸ் ஆர் கோன் பி present, that is நம்ம மைக்ரோடியூபிள்ஸ். அதுக்கப்புறம் சென்ட்ரலி நமக்கு சிங்லெட்ஸ் ஆர் present, so they are all microtubules which are arranged. Okay, microtubules which are arranged. Now moving forward, the central tubules are connected by bridges. Okay, central tubule are connected by what? Bridges, and it is also enclosed by a central sheath. பார்க்கும்போதே தெரியுமே நமக்கு ஒரு sheath இருக்கு and it is connected by your bridges. Okay, bridges are present which are connected to one of the tubules of each peripheral duplex by radial spokes. So just look here. This is the radial spokes. Can you see? இதுதான் நம்ம radial spokes. It is helping it to connect to the center like this. This is the radial spokes. And then there are interduplet bridges. From the duplet there are bridges that are connecting. So those are your interduplet bridges. Okay. And இதுதான் நம்ம central sheath. பார்க்கும்போதே தெரியுமே இதுதான் நம்ம central sheath. So ஒரு circle இருக்கு. Okay, ஒரு circle இருக்கு. அதில் பெரிஃபரியில் கொஞ்சம் ஸ்ட்ரக்சர்ஸ் இருக்குது, சென்டரில் 2 structures are there and they are interconnected with a bridge, and this duplets also have certain bridges, but most importantly they have radial spokes like this which are connecting or which are moving towards the center, towards what students? Towards the central sheath. இது மாதிரி ஒரு sheath is present. Okay, இந்த மாதிரி ஒரு sheath is present. Understood this much? So peripheral duplets by a radial spoke, thus they are 9 radial spokes. Obviously இது எல்லாமே connect பண்ணும்போது 9 radial spokes தான் present இருக்கும். Should I draw it again or just let me just show it to you here only. So 1 2 3 4 5 6 7 8 9. Okay, 9 radial spokes are there. Yes ma'am, understood. Now the peripheral duplets are also interconnected. Okay, they are also interconnected by linkers. Both the cilium and the flagellum emerge from the centriole-like structure. Centriole-like structure called the basal bodies. So on the basal body which we studied in our flagella, they are saying that it is made up of your centriole-like structure or centrosome-like structure. Summary la vandu they have given centrosome and centriole. Paragraph la vandu they have given centriole-like. So adanal you should study both of it. Okay. Okay. My God. Okay, my throat is, I'm shouting. Okay, 1 second students. I will draw one structure for you. Easy structure only. I don't know why students tell that this is very difficult, but it's not very difficult. Okay, first we will draw a circle. Okay, இதுதான் நம்ம circle. Okay, இந்த circle la நமக்கு வந்து குட்டி circles draw பண்ணலாம். So 1, I think if I draw like this it might take 2 days students. So you come after 2 days. Okay, we'll see if we can. Okay, wait. Lot of problems only I have. Solutions are less, problems are more. Okay, you sit here. Where are you getting up and going? Students, one second. Okay, you tell me, how am I supposed to draw like this without a circle? It's okay students, we will go back to drawing it faster than this. Okay, 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 this is the circle. Three, okay, 9 we should draw, 5, 6, 7, 8. Okay, 9 is there, correct? Yes ma'am, almost looks like 9. Okay, now look here. These have, okay, now right in the center, what do we have? We have a central sheath is present. Okay, இதுதான் நம்ம central sheath that is present. Okay, this is the central sheath, and in the center we have the singlet. Okay, இந்த மாதிரி singlet will be there. They are connected by bridges. Okay, they are connected by bridges. Now they all have bridges. Okay, they all have bridges. Anna, what is important for us to remember? They all have radial spokes. So இந்த மாதிரி radial spokes are there. in the radial spokes are moving almost to the center like this, okay. These are our radial spokes and they are all connected to each other. They are all connected to each other with the help of linkers, okay. These are our linkers which are present between them. They are all going to be connected with the help of linkers, okay. This is the general structure. So, when I take a cross section like this is what I am going to get. So, this entire structure axoneme, okay, and this is my peripheral structure. So, these are my duplets, okay. Duplets, how many are there, students? 9:2, no, 9 duplets are there, okay. 9:0 I am writing, and then we have singlets. You know why I am writing like this? Singlets are there, that is going to be 2, here it is going to be 9, and these blue color structures are going to be called the radial spokes, okay, and these are going to be called linkers, okay, linkers, and this is the peripheral structure, student. This is in the periphery, okay, and this is right at the center. This is the sheath, central sheath, understood? So, these are all what? These are all microtubules, okay, these are all microtubules which are going to obviously help in movement. So, peripheral you will have 9 microtubules, okay, adukapram center we will have 2 microtubules, okay, 2 microtubules. Yes, understood? Central sheath, yeah. Got it. Okay. Yes. Axonema is like the core of the flagella, okay. Core of the flagellum, okay, core of the flagellum. So, structure is done. Now, we will see here diagram. See here, students, again this is the plasma membrane, this is the outer microtubule duplet, and then we have the inner microtubule duplet, then you have the radial spokes, right at the center you have singlets, that is your central microtubules, and we have Dyson arms that you do not have to know. Basal bodies are there, we call it as kinetosomes, okay, kinetosomes, and yeah, kinetosomes are there, understood? Yes ma'am, understood ma'am, okay, are you thinking like that? Okay. So, here I will just write this one thing. So, centrosome, we told no, centrosome, centrioles. So, we will have this kinetosomes and your diploplasms, okay, diploplasms plus kinetosomes. That is why we are telling that these people are the ones who are helping in the formation of this, or we can also call kinoplasm, okay, kinoplasm. Yes, okay. Now, difference between cilia and the flagella. Cilia occur through the surface of the cell, while the flagella is commonly found at the end of the cell. So, it is occurring from where, students? It will occur at the surface, okay, this is going to occur at the surface, ana idu vandu end of the cell. The number of cilia per cell is usually very large, while flagella will be 1 to 4. We all know, no, dinoflagellate, max to max 1 flagella or 2 flagella will be there, ana up to 4 flagella they can go, but ana cilia adu madri illa, cilia vandu there will be present, so many will be present, okay, cilia vandu so many will be present. Flagella are comparatively longer and responsible for cell movement, okay, cell move pandradhukku, that is motility kaga nama flagella vandu help pannum. Cilia vandu it helps not just in locomotion, it also help in feeding as well as for circulation. So, cilia, since they are present so many and they are like oar like movement, it will help more of, you know, pushing in the food, and the madri also it will help for not just for locomotion, okay, not just for locomotion. Now, centrosome, very important, okay, very, very important. Centrosome is an organelle usually containing 2 cylindrical structures called the centrioles and the centrosphere. Enna irukku students? Centrosome illa vanda, see here, We have first one centrosome, okay, centrosome is there, and it has what? Centrospheres and centrioles. In the centriole when the right center la irukum, the rest what we call centrospheres. Both the centrioles in the centrospheres lie perpendicular to each other, in the madri. Perpendicular to each other. We will always see, no, they will be perpendicular to each other, in the madri irukum, okay. Now, both the centrioles in the centrosphere lie perpendicular to each other. They are found in animal cell and absent in plant cell, and this is one of the differentiation which we have already studied, okay. Centrosome is an organelle which contains two structures, centrioles and centrosphere, and the sphere is called as centrosphere. Right at the center or perpendicular structure irukku, adudha centrioles, okay, centrioles. Centrioles lies in the centrosphere in a perpendicular manner, okay. Or mari cartwheel madri, I told you, no, cartwheel. Even that other structure looks like that, but Anna, this is more of a cartwheel structure mentioned in your NCERT, okay, NCERT. So, each centrosome has an organization like a cartwheel, each centriole has an organization like a cartwheel, okay, cartwheel. Now, look here. Centrioles possesses a triplet of microtubules arranged in 9+0 pattern. Okay, what is it? 9+0 pattern. Anga vanda 9+2 pattern. Nama Cilia flagella la. Ana inga vanda 9+2 pattern. Okay, that means what? In the center there is no, you know, there is no singlet or anything. It is like this. Okay, one structure is there like this, and from there you have arising microtubules. Okay, arising microtubules. Ana inga vanda enna speciality? There we saw duplet. Ana inga vanda we are seeing triplet. Okay, we will see 1, we will see 2, and we will see 3. So, we will see triplets here. Triplets here, ana center la vandu edume kedaiyadhu students. Okay, center la vandu edume kedaiyadhu. So, we can say 1, 2, 3 illana A, B, C. Okay, like this triplets will be present. So, understood the same thing only, but here microtubule triplets will be present, connecting fibers will be there, ana center la edume present aayirukadu. It's like a hub madri. Okay. The center part of the centriole is proteinaceous and it is called the hub. Just now I told you, it's called the hub, which is connected with tubules of the peripheral triplet by radial spokes made of proteins. So, you can see same like how we studied there, center la sheath irthuchu ange. Ana inge center la or hub irukku. From the hub there is radial spokes which are helping the connection of the entire structure. Okay, and all of this is made up of protein. So, the central part is proteinaceous, even the radial spokes are also protein made. Okay, it is also protein made. So, here you can see students, the centrioles forms the basal body of the flagella and the cilia. They also say centromes and centrioles. Okay, centrosomes and centrioles they say. So, here you can see this is the structure. So, this is the flagellum, basal body also you can see that the centrioles are going to be present. So, centriole structure is clear. Function of centriole, it helps in cell division by forming microtubule organizing centers. What will it form? Microtubules organizing center. Nama vanda prophase, metaphase, anaphase la we study about spindle fibers. All of you remember aster spindle, spindle fibers. So, that these people are going to help. Okay, they will move to both the sides and they will help in that entire spindle formation, so that these people can come and sit in the center. Who? The chromosomes. Okay, the chromosomes can come and sit in the center. Clear? Understood this? Okay, understood this? I will draw this also very quickly for all of you. If I don't draw, then I don't feel complete. Okay, I always tell you this. If I don't draw, I don't feel complete. Okay, now we will draw a hub. Okay, this is the hub. Okay, from the hub, there will be like this lines. One, 2, 3. Okay, like this triplets will be present, and from the hub you should remember students, this blue color lines which I am drawing, these are the radial spokes. Okay, radial spokes. So, I am just labeling radial spokes. Okay, these are the triplets. Okay, these are the triplets. Each of them is what? Your microtubules. Okay, you can call it 1, 2, 3, A, B, C. Okay, and this is the hub. All of this are protein structures. All of this are protein structures. So, like this, it will form like a cartwheel like structure. Clear? Cartwheel like structure. Okay, understood? Any doubts here? Any doubts? Okay. So, you know, centrioles will be there. They are going to help in this spindle formation like this. Okay. They will help in this spindle formation, and they will be perpendicular to each other. Okay, perpendicular to each other. So, this is the entire thing. Okay, entire thing. We are done and dusted with the chapter. If you have any doubts, make sure to let me know in the comment section, and if you enjoyed the class, then quickly subscribe to the channel and smash the like button. And also students, I have also provided today top 30 questions of biology with answer key and detailed explanation. Check it out in the video that I made right now. That is for all the students, because yesterday when I did live, all of you were going so crazy about, ma'am, I'm scoring only 200, I'm scoring only 300, please help us. So for that, I have made a separate video. Make sure to check it out, and in the description box there are 30 top questions from the entire biology, so make sure to solve that questions as well. Okay, explanation is also there, answer key is also there, so you don't have to worry. Then students, I hope all of you loved the class. Please let me know. Since these videos are recorded, you can watch it at your own pace at 2x, 3x, whatever speed you want to watch it. Make sure that you're watching the video. That is very important, because you know, it's very important for you to, like, you know, watch the video and let me know in the comment section, because you know, I'm taking botany after quite a long time. So for me also, the flow to set for you, how you understand, how much examples I can put in, all that will take some more time, because zoology is already, I have been teaching you and I know that, okay, here students will find difficulty, so I have to put examples here. Botany also I am still, you know, in the process. So adanale, it's not like the subject is a problem, but the way how you people are understanding, that is very important for me. So make sure to put your feedbacks in the comment section. If you love the class, you know what to do. So make sure to do all of that, and all the GMA armies, put your attendance in the comment section. See you all in the next class. Till then, take care. Bye-bye.